Hello, good morning. So let's start our second day. Again, welcome for all of you. Yeah. So again, thank you for yesterday. I think we started with a good discussions with with companies, with the policymakers, and today we will have really great discussions also with universities, research institutes, and also funding opportunities from the agencies. Before we start, just some remarks. About our Wi-Fi first, that we will use for the Menti app for the P&H session. And also here again for the attendance that was in these 2 days. And to start, I would like to invite to the stage Dr. Keith Simons from SHV Energy to talk about the R&D current projects from the SHV Energy and the importance of this network.
Thanks very much, Priscilla. Great to be back again, and it was great to see you last night, and I hope you enjoyed the hospitality as much as I did in this fantastic venue. Now, if you think about everything that we talked about yesterday, it all starts with science. It all starts with technology. It all starts with the people and the ideas that come, from this room. So hopefully, ideas that we will foster today by discussing with each other, working with the funding agencies to make sure that we're fully aligned, will be the future technologies, future approaches. Because we've got a long way ahead, and the solutions that we present now are just one potential choice. And of course, it was said a long time ago by the head of the US Patent Agency in, I think, 1901, anything that can be invented has been invented. We know that that's not true, and we need to continuously, sort of push the boundaries of science and technology, because that's what we're here for. So, a little bit about what our view of the pathways in research and development are. Now, one of the challenges that we have, and why we engage so much with universities, is that most people don't think about on-purpose routes to C3, C4 hydrocarbons. So it's, I remember, working with University of Bahia, Santa Catarina, and others last year, and it was that magical moment when there was the realization what's in those golden cylinders is being ignored. I know it seems strange a British guy had to come to Brazil and help you out with that one, but of course that's no different to how scientists and engineers have operated. They focused on the gasoline fraction, the diesel fraction, the aviation fuel fraction. And it's funny, if you go back in history, it was a chemist who actually developed a distillation column to take out LPG in the early 1900s, and there was the birth of the LPG industry. And then I believe that it grew organically. So I think LPG came to South America based as cooking fuel, based on the cylinders that were left over from the Hindenburg. So the Hindenburg was coming to South America. They had all of these cylinders of LPG that were, used. And then, of course, when the Hindenburg was no more, all of those cylinders were left. And it was an entrepreneurial company, still a competitor to ours, that actually, sort of gave birth to that industry based on those important, cylinders. The best ones, of course, are painted golden. So scientists, haven't really looked at C3, C4 hydrocarbons on-purpose manufacture. That's what we're looking to change. So internally, we have 3 different pillars to our R&D strategy. Pillar 1 is our internal R&D programs, where you saw the result of that yesterday. We're closely working where really it's a different type of approach. As an industrial R&D scientist, you just want an answer, yes or no. You want a quick answer. If something doesn't work, that's also a very positive result because you can move on. So we generate ideas internally, we work with trusted parties, looking at those ideas, and we just evaluate them, looking for something which could work. And if it works, you end up, success gets expensive because you start going in more and more details. So we'll always try out an idea, and then of course success comes, continuation, deeper studies to see, is it technically economically viable? I'm an old-fashioned chemist. You take something cheap, you turn it into something more valuable. You try not to do it the other way around because management doesn't like it. Of course, then we screen those ideas, try out ideas, and then we find, well, it's not going to happen overnight. We need fresh ideas. We know there's insurmountable challenges, and the great thing about working with academics is academics love challenges. All we do is we give them the direction, give them the starting point, and then get the focus on looking for solutions that are interesting for us, but also, of course, interesting to academics. I'm a strong believer that academics are fresh ideas, different ideas, but publication of those results is the lifeblood of an academic, and working together in collaboration. And as long as we understand both of our targets, then I see the long-term vision. So the ideas now in the research lab in 20, 30 years' time could be realised or not. But the other thing is that it's the training of the students that come out of that, the scientific thinking, so that when they leave university, they think like an industrialist, because those are the programs that we try to have. Then, of course, any of those ideas, whether it's from our pillar 1 or pillar 2, then SHU Energy, Supergas Brass can't do that on their own. So this is why we had the session yesterday, fostering partnership, because different companies have different skills on that journey to take us there to achieve our 2040 ambitions, that all of our energy products are from sustainable feedstocks. So when I started, it only seems like yesterday, Rebecca, doesn't it? But the state of the art was actually quite well summarised by Eric Johnson, and he had looked at the literature, and these were all of the pathways that he would find. So there's the H3O Hafer process, where you get 5 to 8% propane from the triglyceride backbone. Various commercial companies looking at that. And then there's a whole list of potential processes. So, of course, Fischer-Tropsch, sort of developed over 100 years ago. There's always going to be some propane, butane from that. It's been commercialised now, comes like Fulcrum, Red Rock, looking at this, taking a complicated process and putting a complicated process on it. But of course, can you beat Anderson-Schultz-Flory product distribution? The answer is it's going to be very difficult. But are there ways to try to improve those catalysts? We've looked at it, we've tried, and at the moment it is still very difficult to beat Anderson-Schultz-Flory product distribution. If there's any ideas, we're always interested. Of course, alcohol to hydrocarbons, jet fuel. What's interesting there is that we looked at this process, and then, of course, everyone was focusing on the gasoline, the complicated bit. That was partly our inspiration for Project Katia that we showed yesterday. Of course, people are looking at novel ways, pyrolysis of biomass, again, 2 to 3%. The challenge when you're getting very low yields with a mix of hydrocarbons, it's the separation of the C3, C4s in there. And of course, distillation's incredibly expensive in terms of energy. But if we can go back to the thinking 100 years ago when the LPG industry was born, if we can improve those separation technologies, maybe that's a route. These are things to look at. In gasification processes, people say there can be significant fractions of C3, C4. The fact that there's not a number in there probably says quite a lot. And then looking at biological pathways, oligomerization of biogas, looking at glycerol. People have tried this, but removing those 3 hydroxyls is quite challenging. And then, of course, power to X. So, can we achieve this? But generally, trying to achieve good yields at high technology readiness levels, it's an expensive path. It's an expensive approach. But this is where the scientific literature builds on the scientific literature. We explore these, and then you find something that works after lots of failure. So how do we look at the energy systems? So together with LanzaTech, we're founder members of the Renewable Carbon Initiative. We particularly love this diagram because I think Rebecca said yesterday, it's not about decarbonisation, it's about defossilization. In fact, the fastest way to decarbonisation is defossilization, because we've got all the existing infrastructure. You've got infrastructure in your own homes, all your cooking appliances. We've got the distribution infrastructure, we've got the bottling infrastructure. So if you wanted to have a new energy source that wasn't compatible with that, it slows down the decarbonisation effort. So really think about defossilization as the priority. But we particularly like this diagram and this thinking because we need to think about renewable carbon, recycling the carbon that exists. And the challenge is we're putting fossil carbon, we're burning ancient sunshine and putting the carbon that's in the ground into the atmosphere, and that's the challenge. So we've got biomass, it's our source of renewable carbon. We can recycle the carbon from municipal solid waste or industrial wastes. And then the controversial one is CO2. So of course, direct air capture, taking the CO2 out of the atmosphere, though unless governments around the world repeal the second law of thermodynamics, that's going to be very challenging to do. Though I believe, in Scotland, I think Nicola, will repeal the second law of thermodynamics. I think they're pushing Biden to do it as well. And there's huge amounts of subsidies around, direct air capture. There are plants in Iceland. It is happening, but there you need free energy. As soon as you've got free energy, you can pull carbon dioxide out of the atmosphere to make up for the second law of thermodynamic challenges you have. So that gives us our carbon. And then at the end of the day, where does our energy come from? It comes from the sun. That huge fusion reactor in the sky, gives us the access to solar. Sun still generates wind, hydro, and that gives us our renewable electrons, of which we can make hydrogen if we can't use them directly, and then couple the hydrogen with the renewable carbon, and we can make our chemicals, plastics, and of course fuels, so that we have industrial energy, we can heat and provide energy for cooking as well as mobility. So that's our view, and we share that view with an expanding number of companies. Please look at the Renewable Carbon Initiative. They're doing great work. So, of course, Pillar 1 are internal research and development programs that we normally don't talk about, but then we choose when we will share this information, as we did yesterday. Now, Project Katia, this has been a 3 or 4 year journey. And then I think the 2 years before that we had, lots of failure, but then there's no such thing as failure. There's just learning, and boy did we learn as we tried lots of different routes that in our hands we couldn't improve the performance of those systems in the literature, which is why we look to academics to be much cleverer than ourselves. What was known, of course, that if I'd have read the literature in depth about the ethanol to gasoline process, we wouldn't have bothered, because generally the catalyst fails after a couple of days due to coking. The other thing I would ask the academic community when they publish is, just because something looks good after running for 24 hours, it gives great data, but of course, it's after 100 hours is when you can find out, can you then go to 200 hours, 300 hours? So, just running your reactor for a day, although it's easier to do in an academic lab, doesn't really provide that robust information that you need if you can decide that a process could be viable. And of course, the thing about this process is that the literature talks about ethanol to gasoline. Quite often, they never publish the C3C4 yields. I always imagine that there's people that have discovered fantastic processes to make C3C4 hydrocarbons when they were making gasoline, and their professor told them it was a failure. So always publish even the terrible results, because you never know. If there's a commercial person that says, well, I've been trying to make that. So, this is the challenge, but, just getting people to think differently. That's why we had to do this internally, because no one else was looking at this very important fuel. But I like to think we're changing that. And one of the ways we change that is by sponsoring work at universities, because you never know what's going to happen. So, Jude from Aston, I went to a presentation at Aston University, and I presented to about a dozen academics in the room, and I said, I want to make C3C4 hydrocarbons. And they all listened to me and showed me the research they were doing, and they said, well, can't you make something else? I said, no, we have an entire industry, an entire sector that's delivering hundreds of millions of tons to customers. We need C3C4 hydrocarbons. But Jude, originally from Nigeria, he grew up with cooking gas. He'd been thinking about this before, and he said to me, well, what if we take butyric acid and perform hydrothermal liquefaction? Then you could make propane that way. So we funded a small feasibility study, and we've been working with Jude ever since. And you remember in the previous slide, propane was always a byproduct, 3-4%. The great thing about this work is that it's 95% hydrocarbon selectivity to propane, of course the CO2 from this process as well. But this is it's got huge potential, it's got challenges, but here we're making propane, which in the northern hemisphere, in colder climates, you want propane. Here in Brazil, you've got isobutane, butane in there because it's warmer. But if you're in a cold climate, you want propane because if it's normal butane, then you're okay from 2-3 Celsius. But if you go below zero, the LPG just sits in the cylinder and you don't get your cooking fuel or your heating, so it defeats the purpose. So selectivity to propane is what we're after. It gives high yields. The challenge, it's using platinum and carbon. It's using very high loadings. So of course, challenges in the future will be switching from a battery reactor to a continuous process, and then can we identify earth-abundant catalysts and not platinum? So the other challenge is using butyric acid as a feedstock. It's very smelly. So it's an industrial process. You won't be very popular with your colleagues. So one example, and 3 publications came out of that. So again, it's building up the reputation of the academics. It's getting that information into the public domain. Other work that's using, University of Ulster, in a previous job I worked with the University of Ulster, and they developed microbes that could make sophrolipids that went commercialised by Unilever to make biosurfactants. So when I started, I was looking for partners. I was thinking, well, can you make propane microbially? Now, if you're looking for microbes that can do something, you look for where the products are, because the microbes will be in these areas. So 50% of the world's LPG comes from refining. The other half comes out of natural gas wells. How did it get there 300 million years ago? Or over 300 million years? So is it a thermochemical process, or is it a biological process? So we started looking for microbes to see could we make propane from them. So we took samples from the seabed in the English channels, we cultured the microbes, and then gave them a little bit of nutrient, and then sampled the headspace. And we found in certain cultures that we were getting 70 parts per million of propane. So this was fascinating. The challenge is it's very low productivity, but it means there are pathways. There's pathways from the carbonates that we believe was the starting point all the way through to propane. So microbes, sort of can do that. We've done genomic analysis and we have this data. This is going to be published in the next couple of weeks, so that paper will be out there soon. So if you look out for that title there. So this is a starting point. So of course, companies involved in synthetic biology, such as LanzaTech, can take this information, maybe find those pathways, put them into a different host, and then in the future, you'll be putting CO2, hydrogen into a reactor, and propane will be coming directly out. Of course, we just need to get them much more productive than they are. And maybe it's going to be difficult because if we had 300 million years in the reactor lifetime, you could do it. But, maybe there are clever ways to accelerate that, but I'll let Sean think about that one on his flight home this evening. Then, of course, we've got various other programs. So I was here in August. I must thank you publicly for the hospitality at the University of Santa Catarina in Bahia. And we're starting up these programs. Priscilla mentioned it yesterday, and this is really important. This all started with that small project at Aston and then built in to the reason actually we're here today. So it's amazing what small ideas can turn into. And looking at lignocellulose, biomass to bio-LPG and hydrogen, and then Santa Catarina, and then other important things. So if we think about gasification, we need to get the water content down. We're going to hear more about this today. So strategically important pieces of work. And then also other processes. So in Italy, if you're looking for a PhD or you know someone who wants to go to Italy, the deadline of the 15th of October, if you've got any candidates that want to go, we're fully funding this PhD. And here looking at, can we do CO2 hydrogenation over, zeolitic catalyst, going direct to propane. So funding that with Nancy Arteta at the University of Brescia. So other examples of the community that we're building. Then, of course, but how do we find academics? Sort of myself and Priscilla started off, who people we know. But of course, that's not very, sort of democratic. There might be people that are really clever that we don't know. And then we launched various open innovation challenges. We've run it for 2 years running. The last year we focused on India, asking a simple question, can you generate C3, C4 hydrocarbons from sustainable feedstocks with high yields? So, indigenous biomass, whether it's wastes, whether it's straw, getting people thinking about energy security and using local feedstocks, not shipping hydrocarbons across the world. So I think because we did this during lockdown, so people were looking for this, attending the webinars. So we had 27 entries for this competition, and then various proposals, and then we narrowed it down to the 5 that have continued with this research. And of course, we've got 2 winners from our Indian competition. We'll be announcing that soon. So again, fresh ideas, but going far and wide looking for these ideas. Then the other really important thing, and one of the reasons we're here, is engagement with the scientific community. So if we think about, it was a year ago, we thought wouldn't it be a really good idea to do a webinar? And we had, similar to yesterday, we had the government, we had the industrial representatives, the academics, and I believe this was the first technical meeting thinking about bio-LPG. If it wasn't the first, it was certainly the best at that time. I think we've now got to the next stage. And of course, we had people from all over the world viewing in, generating that buzz, and then some of you will have found out about bio-LPG by attending that lecture. And of course, that's led to today. So really important. And at SH Energy, we're actually very open about a lot of our R&D programs, and then share at other times. And that's led to us being here today. So again, thinking about now, we're making, looking at societal, some problems, and then we see bioLPG as that solution. And then, of course, I mentioned we can't take ideas in the research lab and think we've solved the problem. That's only the beginning, and then we seek industrial partners, whether it's Atlantatech, Q Technologies, UGI, to get there and achieve our objectives. And Priscilla's hovering, so that clearly that isn't there. So take-home message: we're always looking for new partners, fresh ideas, whether it's a publication to add to the scientific knowledge or potentially a commercial pathway. We can fund some, but not all projects, and a feasibility study sometimes, but often, leads to something much larger, is our experience. And then remember, the global market for bioLPG is perhaps more important than sustainable aviation fuel, but you wouldn't believe it from the funding available in both areas. So finally, please join us in advancing energy together. Thank you.
Thank you, Keith. Sorry for that, but probably I couldn't open for the questions now because we are a little bit delayed, but I really recommend if someone has some questions, just connect with Keith, mainly during the coffee, right? Because the main ideas is born from the coffee time. So in order to follow our agenda, I would like to invite to the stage Dr. Karen Pontes, that will talk us about the biomass treatment for the gasification. Karen, the floor is yours. So this is the Menti QR code again, if someone needs.
So good morning. So I'm Karen Pontes. I am professor at the Chemical Engineering Department of the Federal University of Bahia. And first of all, I'd like to thank SHV and Pecasbras for inviting me to be here and to give this talk for you. Well, I'll talk about the biomass treatment to improve gasification. Well, we can say that the biomass is a renewable source of carbon and hydrogen because it stores the solar energy in carbon bonds. And then the biomass plays a key role in this energy transition towards a carbon neutral economy. And then there are several pathways to convert biomass into renewable fuels. And one of these pathways is gasification. And the gasification is a thermochemical conversion of biomass into this mixture of gases called syngas, which is composed by hydrogen, carbon monoxide, and CO2, which then capture again from the biomass during the photosynthesis. And then we say that this is a carbon neutral process. And in the gasification, there are several zones of operation. The first one is the drying zone, where the biomass where the moisture content of the biomass is reduced. And then we have the endothermic reactions and the pyrolysis and reduction zone. And here in the combustion zone, we use the biomass to provide heat to the other zones of reaction. To drive the heat in the combustion section to provide heat to the other endothermic zones. And then the higher the moisture content here of the biomass, the higher will be the heat required in the combustion. That means the higher is the amount of biomass we'll need to provide that heat. That means instead of driving the reactions here, we'll use this heat to dry the biomass. So that's our point. And then, as you can see here, what happens with the moisture content in the biomass. This influence here the heating value of the product, of the bioproduct. And then you can see that, when you increase the moisture content of the biomass, the heating value of the biomass decreases because, as I mentioned, instead of driving the reactions, the endothermic reactions of gasification, we will dry the biomass with that heat. And besides influencing the heating value of the biomass, we also has the scenario here, the syngas composition also changes. And that means, for instance, this CO2, that means this is the moisture in biomass when it increases. So the CO2 content in biomass and the water content increases. So this is the complete combustion. Whereas the CO2 content decreases. So these are related. Yeah? So we decrease the CO2 content and the partial oxidation, which actually would have to happen. And then we can say that the moisture content in the biomass reduces the energy efficiency of the gasification and also the syngas composition. Therefore, we need to remove the moisture content of the biomass to improve the process. And usually, the wood biomass presents 50 to 60% weight of moisture content. But ideally, it should reach 12 to 15%. Because actually, we still need water and degasification also to target the composition of the syngas. But then, we need to reduce, we need to remove the water content and the moisture in the biomass. And therefore, we need to dry before the gasification. And some common process that are used is a rotary dryer. And this is, it can use hot air or steam. And then we need heat. And therefore, these processes are very high energy demanding. So we need to remove the free water, the bound water that is also connected through hydrogen bonds and the biomass. Therefore, we need to find new energy efficient methods to reduce the moisture content of the biomass. And therefore, we are here. And in this journey together with this SHV and as Rebecca talked about yesterday, the DME. So we are also together in this journey. So, and then our goal together with SHV and Supergasbras is to implement the watering technique using DME. I'm going to discuss more about that. So this technique for removing the moisture content in the biomass, and then investigate its impact of the dried biomass on the gasification and on the energy of the process and also on the syngas composition. And then we come again to DME. And as Rebecca mentioned yesterday, so this is very similar, the properties of DME and the LPG. So we can replace we can use 30% of DME and LPG using the existing infrastructure. But then what is DME? So DME is the dimethyl ether. This is the simplest ether. And this is a gas at normal temperature and pressure conditions. But then it can be easily liquefied at 5 bars at the normal 27 degrees. It can be liquefied. And then other properties that are important here are that DME forms hydrogen bonds with water, as you can see here, and it presents miscibility and molten solubility with water. And that makes possible to extract water in the biomass using the DME. And then some other important properties of the DME, so some properties which make pressurized DME an appropriate choice for the extraction of natural compounds are the chemical inertness of DME, the high affinity for oil compounds, and that means that besides extracting water, we can also extract some oil components. And some of these oil components might have some added value, so we need to check, we need to characterize. And as I mentioned, it has partial miscibility with water. And very, very important is that we can easily remove the solvent, the DME, via pressure reduction. That means we can recover the solvent, the DME, after the watering and then recover it again. So this is the concept of the DME and the watering technique. So we have here the wet material where the white color represents the water, the moisture content in the biomass, and the black color is the biomass, and this is the liquefied DME. Then, if you put in contact, the DME in the biomass, in the wet biomass, the DME will enter the pores of the biomass, and through the hydrogen bonds, it will extract water and maybe some oil components. And then afterwards, we'll get the dried biomass and the liquid phase, I mean, the water and DME. And then if we reduce the pressure, we'll recover the DME and get the water that was removed from the biomass. And this is the sketch of the experimental setup we're going to implement to install at the university. So this is the storage vessel of DME, and then the column of biomass where we're going to extract the moisture content, and then the separation vessel to recover the DME, and a trap column to get some possible water that was carried together, and then we recover the DME to use it again in the process. As I mentioned, through pressure reduction, we can recover the solvent. Well, and then the literature reports the energy consumption of the wood chips biomass, the watering process. We can see here for different species of wood that the liquefied DME presents a much lower energy demand than the oven, which is usually in labs. And actually, this process here uses heat, as I mentioned before. So heat is the driving force. But now, when you get to the DME, we don't need heat, as I mentioned, use pressure, and then the energy required here is actually used to recover, to compress the DME, to use that again. So therefore, we want to achieve this low energy demand. And then here are some examples reported in the literature. We can go from vegetable biomass to waste, water, and oil sludge. We can see the raw material here and the solid product, the dried product, and the liquid phase here. You can clearly see the oil that was contained in the biomass also, that was extracted together with the water. And there, the vegetable biomass, for example, pericarp of mangosteen, and we'll have the dried biomass and the liquid also extracted. And here you can see the extraction efficiency of different matrices from literature and fixed bed and multi-batch extraction systems. So this extraction efficiency against the DME in the water amount, I mean in the biomass. And I highlighted there the cattle manure and the sewage sludge. They are in this upper rectangle here, so we can reach up to 100% of the watering, of the efficiency of removing water. And in the case of microalgae and vegetable biomass, it's also high, roughly 85% or even more. And then that shows us that the extraction efficiency in coals, sludge, sediments, and manures was superior to biomasses, which indicates a higher share of physically bound water, free and physically bound water. And that means that we started thinking about using biomass, as we saw yesterday. So Brazil has a great potential of using residual biomass as agricultural residue. But then, when you see these results and the discussions from yesterday, so we want to move forward, and then also we'll include in this search here, so the forest, besides forest crops and residuals, we include the seawater sludge, RDF, so refuse-derived fuel, and animal residues. So we are going to compare how is the DME dewatering using vegetable biomass and these other waste materials. And we can also use algae, for example, that was already represented in that graph. Well, and this is some pictures of our lab. And we have some years, not only me, we are a group, yeah, so we have some decades of experience in producing biofuels. So we have a pilot plant for gasification, also there, upper there, and a pilot plant for biodiesel. There, and the top, in the bottom, is the torrefication pilot plant. It's another pretreatment of biomass. And then we are going to compare also the results. How is the torrefaction, the torrefied biomass, against the DME dewater biomass. But in the case of this project with SHV, we're going to focus still be in the lab scale. So we are, as I have shown the apparatus, the setup, we are going to implement that. And then after the DME dewatering, we are going to use our setup for gasification. So this is just a lab scale gasification. So before going to the scale up. Well, and then this is our vision, our, how can I say, our activities in this integrated biorefinery. So as I mentioned, we have some years of experience in our group doing gasification and synthesis of some renewable fuels. And here, for instance, if we get to the residual biomass and also putting together RDF and other waste and municipal waste, for example, and then we will do the dewatering. Also, this is the approach here. But then using the dewatered biomass, we want to improve the efficiency of the gasification. And then we go to the synthesis section, where you can synthesize some biofuels. For instance, the renewable DME, which can be used in the process again. So this is an integrated biorefinery. And then we can also use, as I just showed you, microalgae, for example. And then we can use the DME dewatering to dry the microalgae. It has, as I mentioned, almost 90% of efficiency of removing that water. And we can use the solid biomass to do the gasification, or we can get the oil and do transesterification. So I have shown you that we have also some research in the biodiesel production. So we can do the transesterification, and to do the transesterification within methanol that can also come from this integrated biorefinery. And the glycerin that can be used for producing biopropane and biobutane. And last but not least, if we have CO2, and then instead of having just a neutral process, carbon neutral process, we can get the CO2, capture the CO2 and storage that to get to a carbon negative process. So this is our idea, and our activities are in this concept here of this integrated biorefinery. Well, and then I'd like to thank you for the attention.
So great presentation, congratulations, Karen. So sorry everyone again to don't open the questions because as we started a little bit delay and we are also in live stream, we decided just to don't open the questions now, okay? But feel free to contact Professor Karen, and also if we receive some questions by Menti, we can share with Karen and to reply first before. After, sorry. So to follow our agenda, I would like to invite to the stage Professor Dr. Bruno Carcioff from University of Santa Catarina to talk about biotech to produce biofuels.
Thank you, Priscilla. Good morning, everyone. I would like to thank you for the invitation. This really nice workshop. Thank you, guys. Supergasbras, HV Energy for this. The topic today is quite broad. I'm not going to explain everything about biotechnology because even I need many books to get some details on that, but we're going to talk about what we have in mind. Mainly, in this case, let's say the results from our first year in the partnership with Supergasbras, SHV Energy. So we worked together in that Samba UK project, and then we came up with some ideas that we are going to share. I'm from the Federal University of Santa Catarina. I'm representing a group of professors and researchers that accepted in such a way this challenge to go to the production of biofuels. I think I got the wrong way. The green one. Oh, here. So this is the agenda. I will start with this overview about the production. And as Karin already told you, of course, biofuels are fuels from biomass, and we have many options to obtain biofuels from that way. Our interest in the biotechnological routes, even there we have many options, specifically for this project, our goal is to obtain biopropane, bio-LPG. So all these challenges that I'm going to share with you, yes, this is what I'm going to share. Challenge more than results still for now, is to obtain the bio-LPG from the biomass. As Dr. Keith started this morning sharing with you, the literature is presenting some nice data about how to obtain bio-LPG if you have butyric acid through catalytic process. So in that way, considering that's not a biotechnological route, I'm not addressing the details here, but this is also something in our, let's say, our goal, considering the partnership with SHV Energy, Supergasbras, guys from Aston University. And of course, we have a challenge still there to make it an industrial process. So, as butyric acid is a precursor, what we need is a bio-based butyric acid. Butyric acid is already used in many industries, but indeed, it comes from fossils. So the idea here, the proposal, is to make a different route based on renewable lignocellulosic biomasses to obtain the butyric acid. And specifically, in our case, we are looking for bioproduction. That means using biotechnological routes to obtain that important precursor. Let's think about a little what we have when we talk about the biomass. What is a biomass? In general, the lignocellulosic biomass is composed by lignin, cellulose, and M-cellulose. Cellulose and M-cellulose can be converted to sugars that are fermentable. So this is the main idea when we discuss using lignocellulosic biomass to obtain butyric acid. Is to, first of all, open the structure and obtain these sugars. Some examples, of course, you know, we have discussed it a lot. In Brazil, the agricultural waste is really abundant, and sugarcane, soybean, corn, wood are good examples of biomass that can be used, and you are investigating, screening these more important sources. Looking again to the structure of the biomass, so we have the cellulose, the M-cellulose, and the lignin together. Before that we reach the sugars that will be used by the microorganisms that we will talk further, we need to open the structure. So normally we call it the pre-treatment, open the structure, separating cellulose, M-cellulose that are polymers that we need to break down from the lignin. Yeah, so different options we can find, chemical, physical, biological. We can combine them, but indeed what we are looking for is a procedure, of course, always low energy, low costs, but specifically that can increase the delignification. That means separating the lignin, but also avoiding inhibitory substances that can impair the fermentation. And something important that I would like to bring to you, each biomass will require a specific process because the structure is different. This is why we need to adapt and think about each case and what you want from that. After that, we need to break down those molecules. We call it saccharification. That is indeed hydrolysis, normally using enzymes. So we need to think about different enzymes. So we have here another process that, let's see, here these enzymes can be inhibited. So we need to take care about the content of sugars, phenolic compounds, and also we need to think about recycling, optimizing the process to make it viable for industrial process. After that, here I'm not going into all these details because Priscilla told me I have half hour. Is that 1 hour? Is how much is? So of course the biorefinery can produce different compounds here, working with the biomass, the pretreated or the hydrolyzed. But in our case, what we are looking for is to produce organic acids. And we can have different routes, but the most efficient seems to be working with the sugar. So this is what we want to obtain from our biomass. Specifically for the production of butyric acid, again, I'm not going into these details, but we have some options using microorganisms. In this case, example is the genus Clostridium that has this metabolic pathway, the ABE, so-called ABE fermentation. ABE fermentation means that at the end we will obtain these main three compounds, the A, the B, and the E, but during this pathway we also obtain some intermediate molecules. And in this case, what you're looking for is to move the system in a way to increase the yield of butyric acid. So this is the strategy in the fermentation process. That means process parameters, that means different microorganisms and combining everything to really go in that direction to increase the yield of butyric acid production. After we have the sugars, or indeed here we have the process, the fermentation process, including the saccharification. In our group, we are proposing to evaluate 3 different arrangements. This first one means that, for example, we can use the enzymes before and then move to our reactor where we add the microorganism. But we have other configurations in order to maybe optimize or to achieve better feasibility for the process. So, for example, we can add the enzymes and the microorganisms in the same reactor. In this case, for example, while the microorganism is consuming the sugar, it can avoid the inhibition by sugars of the enzymes, and we can optimize that. In another configuration, the consolidated process, what is different that we can add different microorganisms, some that synthesize in loco the enzymes that will break down the molecules producing sugars, and others consuming the sugar, so the fermentation and the enzymes production are done at the same time. It's not the end still. Now we have a production of we want. Someone were discussing yesterday. After we have the mixture of everything, so we need to separate the downstream process. We need to obtain the most purified content in butyric acid. For example, we have many strategies for this separation process, from membranes, adsorptions, and so on. We believe that in this sense, it is still a work, it's still a challenge that we need to overcome. And again, low cost, low energy, but specifically the selectivity of the process. And again, a big challenge here. So we have the butyric acid, but we need to take it out from that mixture. Briefly, I will share with you some data on the production of butyric acid through fermentation. And the idea here is not to necessarily go to the data, to the numbers, to the yield or the productivity, but to discuss that depending on the biomass, depending on the reactor configuration, of course, depending on the pre-treatment, we can have higher or lower yield and productivity. Because if you have high yield, but it takes so long, it could be a problem. So we need also to have nice productivity. That means the amount of acid produced in an hour per liter of our reactor. So this is so important in terms of thinking about industrial process. Here, the main factors that you are taking care when you are thinking about this production. So the pretreatment and the concentration of fermentable sugars, the fraction M-cellulose will affect the amount of C6, C5 sugars there. The inhibitors, we are not talking about that much about the detoxification process, but this is so important in some situations, depending on the biomass you are using. The supplementary nutrients, the process mode, batch, feed batch, the time, and also, of course, the separation process. So, trends and challenges. Again, my talk is basically challenge, and this is what we are developing, and this is why we are working so hard to find solutions to make it easier. I have this arrow that means that we are getting there, but very important here, each arrow is connected to each other. This is so important to have in mind. So depend on the pretreatment and the challenge we have here, we discussed it a little, it will generate a different structure. And after that, we have the enzymatic process that is impaired or facilitated by the process, but also the previous steps. So we need to think all this together to find the process, develop it, to understand the kinetics, and so on. Next step, I mostly skip it during the presentation, but if the lignin is not removed, but it's broken during the process, some phenolic compounds can be present, and it's a problem. It is a problem for the microorganisms. So maybe we need some detoxification process depending on the biomass. Of course, the fermentation, that what we want is to know exactly how to manage and to increase yield and productivity, and at the end, of course, high recovery, high specificity in our process to reach what our goal. Butyric acid, that's not the end still, because then we have the conversion to the bio-LPG. So I would like to thank you again. I'm really in a pleasure to be here discussing all these things with you. Here we have a QR code for the reference if you need to see a little bit more, so some that we are suggesting, and open for questions if you have time now. If don't, we can discuss further. It's always a pleasure. Thank you, everybody.
So in fact, I think we have time for one question. There is some questions. No? So thank you very much, Bruno, again. So follow our agenda. I also would like to invite to the stage Professor Dr. Vânia Pazza to talk about the biorefinery of Macaúba into biofuels.
Good morning. My name is Vânia Pazza. I'm the professor from Universidade Federal de Minas Gerais, UFMG. I'm a chemical engineering, and today I will talk about the biorefinery of Macaúba, and I will present some results that we obtained in our lab over biofuels and renewable gases. Firstly, I'd like to thank Priscilla for this special invitation to be here in this special place with a special person to think in our future. I'm very glad, Priscilla. It's very nice. Well, I'm the coordinator of this field lab in our university. I will give some information about our work because you need to understand what kind of work we have done. Our lab is a multi-user laboratory. Then we work to meet the demand for companies in Brazil, for the researchers, students, and the government. We have almost 22 years of experience working with fossil fuels and biofuels. And we are considered an innovative catalytic thematic environment because we have investigating many kinds of processes to produce biofuels. I'm the founder of the lab, and our lab is in the Department of Chemistry of our university. As you can see, our lab can have 3 kinds of projects. We work with research activities, we work with special service for companies, and in this case, we can attend the demand to certify the quality of all kinds of fuels in Brazil. And also we have a human resource training program with ANP. ANP is the national agency in Brazil for petroleum, natural gas, and biofuels. And in this case, we prepare students for the market. I would like to highlight that our lab is accredited by the Inmetro, which means that you work in compliance with the requirements, technical requirements of the standard ISO 17025, which means that we offer a service with international quality standard. Well, sorry. As now I will talk about Macaúba. As you can see, Macaúba is a green gold, especially because the properties of the plant and the diversity of products that you can produce. Macaúba is a palm tree with natural occurrence in Brazil, especially in my state, Minas Gerais. And it can be pointed as a promising fuel, feedstock for fuels, especially due to its high productivity in oil. Macaúba can produce about 6,000 kilos of oil per hectare per year. It's very similar to palm tree, but there is an important difference. Macaúba has more sustainability because this plant doesn't demand special climate, special soil, and can recover degraded area. Francisco from Solium will talk more about this plant, maybe after lunch, I'm not sure, but he will learn more about this plant. And we think that it could be one interesting alternative to provide feedstock for sustainable aviation fuels, green diesel, and gases. Here we have a fruit of Macaúba, and in the fruit we have 2 parts of this fruit that can produce the oils. We can have the pulp oil from the mesocarp. In this oil, the main compound is the oleic acid, and this oil is similar to olive oil. And the second oil is made from endosperm, and the main compound is the lauric acid. It's a light oil. When we process the fruit to produce the oil, we can separate some wastes that can be used. For example, here, when we produce the pulp oil, you have the cake, the pulp cake, and you have the almond cake. These cakes can be used for animal feed or to produce second-generation ethanol, or now we are working with bio-oil and bio-crude. Other possibility is to convert the endocarp, this black piece of the fruit, into charcoal or activated charcoal for industry. And in this process, the bio-oil is a co-product very important too. The pulp oil of Macaúba is very suitable for the biodiesel production. Petrobras has been using this oil in some refineries in Brazil, and we have already worked with this. And in this case, we are showing one project that we developed with the CNHI Industrial company from the Fiat Group, and we prepare a biodiesel from Macaúba for tractor engine, and the result was very nice. In this case, we had a problem because the oil was stored for many months, and the acidity improved a lot. Then we had many problems to convert it into biodiesel, and the alternative that you provided is to reduce the acidification with esterification with glycerol. Then for this reason, it's very important to pay attention in acidity to convert it into biodiesel. But if you want to work with sustainable aviation fuels or other biofuel, biohydrocarbons, for example, is not one important problem. Here we can see some results of our lab, obtained in our lab, when we produced Macaúba biogasoline, green diesel, and biokerosene. Here we have 4 fractions and their chromatograms. In the bottom you can see the Petrobras fossil kerosene in the black color. Each peak is one molecule. Then as you can see, our biokerosene is very similar of the fossil kerosene. And in this kind of process, you used catalyst, waste catalyst, and low hydrogen pressure, and biopropane was one important co-product. In our lab, we are always worried with price, and we are always doing things to reduce cost, especially using waste for the biofuels production or biocatalyst preparation. As example, in this case, we prepared the catalyst using the endocarp of the Macaúba to produce SAF, sustainable aviation fuels, and biokerosene. The result was very nice because the efficiency was 98%, and the quality of the product was very high. And as you can see, we reduced the cost and we improved the sustainability of the process. These works have already been published in this paper. Here we have 2 other papers in which we present our results using different kinds of catalysts. I would like to point out that to prepare different catalysts is one expertise of our group, and I think that we can use to help other groups here in other projects. Another, we have a new result, because I was worried because I came here and I have to say something about gases. Then now I'm paying attention only in gases. I changed my mind now. And now we prepare another catalyst, and with this new catalyst, we obtain HVO and SAF, propane, but now we observe the presence of biomethane. Then I think that we can produce another gas if you change the catalyst. And here we have one excellent result. In this case, we prepared the bio-oil from macaúba wastes, and we tested two kinds of catalysts. In the catalyst A, we obtained, for the upgrading of this bio-oil, we obtained biogasoline. In that picture, you can see the sample, and this sample has a heating value higher than 40 megajoule per kilo, which is very nice for a waste, a solid waste. And in the bottom, you can see that we use in catalyst B, we produce SAF and biogasoline. Again, we observe the presence of biomethane, methane, and we think that we can produce biodimethyl ether changing this catalyst. This is the next step that I will do in our lab. And here we are showing another research in which we used another process. Here we use the solvent to liquefy the solids, and we obtain a biocrude with high heating value and the biochar. And this biochar can be used for agriculture purpose. It's a good result too, because you can do this in a small time. A few minutes, we can convert solids in that kind of oil, renewable oil. I like this process, but it's necessary to use high pressure. But the temperature is low, and about 50 minutes, everything is finished. And here we have other results of our lab, in which we studied the production of hydrogen. And in this case, we prepare hydrogen using cellulose and zinc oxide catalyst. And during the process, we obtain nanostructures of zinc and nanowires, nanoflowers, and many nano things using this process. We have a patent with this, and we are beginning another process to produce biohydrogen using bio-oil and biocrude. And we have a big project to produce biohydrogen from ethanol. In this case, we are doing with a partnership with automotive industry. And now I would like to show you one interesting result in which we tested two different catalysts in the pulp macaúba oil. And in this case, in this option here, we use a catalyst that we produce only liquid process. And there we have another kind of product, a semi-solids. And when we analyze this, we observe that we produce biokerosene hydrocarbons, green diesel hydrocarbons, and biolubricants. We think that this result is very important because lubricants is very expensive. It can improve the revenue of this biorefinery and to reduce the cost of the biofuels. Here we can see other projects that you have to produce bioresins. In this case, we produce biofoams using the solid residues for thermal insulation and package market. We want to use all part of the macaúba tree and to aggregate revenue in the all process. Finally, I state that Macaúba is a promising alternative for the planet decarbonization. I'm sure. I have working with about 10 years with this palm tree. And I can say that we can produce liquid and solid, as you can see, biokerosene, biodiesel, green diesel, biolubricant, second generation ethanol, bio-oil, bio-crude, bio-charcoal, biochar, and gases, biomethane, hydrogen, dimethyl ether, and others that we will do in the future, I hope. Thanks for your attention, and thanks for the invitation.
So probably we have time for one question before to Keith.
Thank you very much. Fascinating. So there's huge potential for Macaúba oil. I think you've convinced us of that. What are the commercial barriers for developing Macaúba plantations in Brazil? Is it there's that commercial challenge? You're talking to plantation owners that could have those forests of Macaúba, sort of palm trees. Is that happening, or what are the barriers?
I don't know if I understand, but at this moment we have Macaúba only in native forests, but we have one important company, Soleá company now. The Soleá is changed the name, and we have a Solium. Francisco will present, and they are investing and planting a lot of Macaúbas to change the world, to change Brazil. And I think that we will have many forests in maybe 5 or 10 years. But now we can use the samples only for native. But in 5 years you can not cut, but you can obtain the fruit because the plant is a perennial plant. Okay.
Good morning. Thank you very much for the presentation. Brilliant. We have a question here from the Mentimeter. What's the energy balance for producing Macaúba biofuel, and how much energy do I spend versus how much energy do I have at the end?
Well, it's difficult because we don't study the energetic balance. We didn't study it yet, but as you can see, we have high production of oil. There is nothing that can produce more oil than palm tree. Nothing. And if you want to produce liquid fuels, you have, in my opinion, you have to work with palm tree. But the problem is the sustainability. And here we find one palm tree that will have high sustainability, can recover degraded area, can recover water source, and can produce oil with high amount in this small area. And I think that this is very nice. But if you consider each product here, we will need to, at this moment, we want to work in pilot plants. We have one that will begin now. Then we will do this kind of energy and mass balance to answer you more maybe in the next event.
Thank you very much, Professor Vânia. So, Now to talk about the pyrolysis of plastics to produce biopropane and biobutane, I would like to invite to the stage Dr. José Carlos Pinto from University of Rio de Janeiro, Federal University of Rio de Janeiro.
Good morning, everybody. This is a new format for me, so I hope I will not get too confused with the slides. Okay, so the title is somewhat different from what you saw in the program. I removed the word or the expression bio on purpose because most plastics nowadays are not produced using bioresources, but the whole field is moving towards this direction. So please consider that in the near future, we'll be talking about bioplastics. I want first to thank Priscilla for the invitation, also Priscilla and Keith for the very interesting technical discussions. As a matter of fact, we tried to follow Keith's advice, and I will try to present some general information about the production of plastics and gases in general. And obviously, I would like to thank the colleagues, the students, partners who are responsible for most of the figures that I will present. Sorry? It's okay. It's just to provide a message that the pandemic is still outside. So the outline of the presentation is as follows. I will present some general information about the production of plastics and gases with the objective to see if there are justifications to produce gases from plastic residuals. Then I will propose the definition of the problem so that we can provide the context of the work that we are doing in our group, present some results that we obtained in our lab very, very, very quickly, and then present some final remarks. You probably have already seen these pictures or pictures like that in the past. Basically, it shows that illustrates the explosive growth of the plastic industry. Actually, this is a problem of our 21st century. We must solve this problem. It's estimated that the society will have produced more than 4 billion tons of plastics until 2030. Please keep that number in mind. And it's estimated that only 6% of this whole amount of plastic has been recycled somehow. And unfortunately, more than 70% of the plastics are now available or lying on landfills, and worse than that, on the environment. So it's a major problem that our society faces now. For comparative purposes, I'm showing here the global demand of natural gas, and we see here that our society is energivorous. We consume high amounts of energy, and natural gas corresponds to only 25% of the energy that we are consuming nowadays. But assuming, using a very simple calculation, that all that plastic material could be somehow converted into natural gas, all that 10 billion tons of plastic would be sufficient only for 1 year's supply of natural gas, which means that it does not make any sense to use the plastic residuals to produce gas. When we are talking essentially about methane for production of energy, generation of energy. But when we talk about LPG, the scenario is very different because according to the information that we can obtain in the web, the global demand is around 300 million tons of LPG, and about 100 million tons of this LPG is used by chemical plants. If we keep that number in mind and we compare 100 million tons per year with 10 billion tons of plastic waste that are available nowadays, there would be sufficient plastic to satisfy the global demand for 100 years of LPG. This clearly shows that, well, it may make some sense to produce LPG from this plastic waste. Besides that, given the explosive growth of the plastic business, we are still producing 400 million tons of plastic every year, which means that a single year of plastic production is sufficient to guarantee the production, assuming that it can be completely converted into LPG, 4 years of the global demand of LPG products. So I will skip this slide. Well, just to show you very quickly that the scenario is not very different in Brazil, so that we could assume similar figures, similar numbers for our country here. So we can provide some very short conclusions at this moment, that using residual plastics for manufacture of sustainable LPG makes sense. And there are plastic weights that would be sufficient to keep the business running for a very large number of years. And at least in principle, recycled LPG should not compete with other sources of renewable and sustainable types of energy, as we saw here, because the amount of energy that the society consumes is much higher than that. So let's go back to the definition, to the problem definition. In our group, we are pursuing the general objective of keeping the carbon atoms and the hydrogen atoms circulating in the chemical chain. So the idea is that we should avoid talking about waste or using landfills as the final solution for the position of the product waste that our society produces, and use the wastes as additional opportunities for manufacture of other products. This is the well-known scenario of the circular economy. And I must say that plastics are champions in this regard, because plastics can be recirculated, recycled in the chemical chain through many different ways. They can be reused, recycled mechanically, and even when the plastic pieces are not useful anymore, they can be recycled chemically into the chemical chain again. And this is the work that we are doing in our group. When we talk about the use of biomass, the scenario becomes very, very interesting for plastic materials, because now we have an additional opportunity to keep the carbon atoms in the solid state. If we can remove the carbon dioxide from the atmosphere, produce plastics, and keep it recirculating, recycling in the chemical chain. So we definitely see that the manufacture and use of plastics, although the society does not have a very good opinion about plastic materials, but in my opinion, this is completely wrong. We see that plastic materials in general are friendly for the environment, and they can provide very good opportunities to capture carbon dioxide from the atmosphere and keep it circulating in the chemical chain. And this is the work and the idea that we are pursuing in our group. So a couple of additional short conclusions is that the main challenge for sustainable circular chemistry is keeping the carbon atoms circulating in the chemical chain, and plastics provide many advantageous characteristics for development of circular technologies, eventually even to produce LPG materials. So, some quick results from our lab. We have been working in this field for quite a long time. I invite you to check some of the papers that we have published recently regarding pyrolysis technologies, life cycle assessment of plastic materials and technologies, modeling of polymer production and polymer degradation, and so on, and some books that we have written talking about these technologies. In our group, we are ready to analyze the recycling, particularly the pyrolysis and gasification of plastic waste in different scales. We have some very small lab scale reactors. We have recently acquired a pilot plant reactor that allow us to produce 10 liters of recycled materials per batch, and we are now working in a project with Petrobras to build a pilot plant in Cidade Universitária, Ilha do Fundão, in the Federal University of Rio de Janeiro, to process 1 ton of plastic residuals per day, which is the amount of plastics that reach the shores of Cidade Universitária every day in Ilha do Fundão. We produce things like that, different streams with different compositions using municipal solid wastes, marine waste and debris, and so on. But we are particularly interested in producing gases and light fractions for the chemical industry. We are assuming that somehow the final process that will be used to recycle these plastic wastes will include different steps, certainly milling of the waste, pre-treatment for removal of chlorine and oxygen, particularly carbon dioxide. Then we, the material or the feedstock goes through the heart of the process, which is the pyrolyzer, and eventually the gaseous stream that is produced in the pyrolyzer is modified and cracked additionally for tuning of the properties of the final stream, and at the end we have some sort of separation through distillation or flash vessels. This is just to illustrate that we can tune the characteristics of the products that we produce during chemical recycling. We can produce, for instance, almost 90% of liquids, but when we combine with certain catalysts, we can produce as much as 78% of gases. Actually, that 70% can reach numbers that are very close to 100% because we can recycle the heavier hydrocarbon molecules that are produced to produce additional amounts of gas. This is not considered in this slide, but it's important to show that we can produce large amounts of gases. And when we look at the gas compositions, we see that almost 50% in weight basis regard the sum of C3 and C4 streams, which is exactly the fraction that we are discussing here in this seminar, in this meeting. This is an interesting slide because we see here carbon dioxide, and what we are seeing here is the effect of the pretreatment stage and the successful removal of carbon dioxide, which is a problem for processing of gaseous streams in some petrochemical units and refineries. And it may be very important to remove carbon dioxide from these gaseous stream, and this slide is showing that we can definitely do that successfully. Following Keith's advice, we performed some very simple calculations, assuming that the gaseous stream should be separated into C3 and C4 streams in accordance with the Brazilian specifications, and we decided to ask the following question. How much energy would it be necessary to separate the C3 and C4 components from this gaseous stream? And what we observed in many simulations is that we never needed more than a few percent of the total energy content of that stream to separate C3 and C4 in accordance with the Brazilian specification standards, which means that from these very simple calculations that this operation would be economically and technically feasible. There is the problem of the scale. We have been discussing this point with Keith, but it seems very clear that we do not have an economical and technical constraint for production of C3 and C4 streams from this mixture. So the distributions, some very quick conclusions again. The distribution of gas compositions can be broad, although we can have as much as 58% of C3 and C4. And this is due to a particularly reaction step named random scission, which means that the carbon atoms, the bonds between the carbon atoms are broken at random, and this leads to random production of molecules of different sizes. It would be very interesting if we could somehow control this particular reaction step, a technology that is nowadays usually called as plastic to monomer technology. And as we said, purification seems to be technically and economically feasible. We are studying this plastic to monomer technologies in our group, and this is just presented here for illustrative purpose. After adding a certain catalyst, unfortunately, I'm not allowed to tell you what the catalyst is at this particular moment, and I'm not allowed to tell you the reaction conditions, but it is much milder than the user temperature conditions used to perform the pyrolysis reactions, and we can increase very significantly both the amount of gas and also the C3 and C4 content of the gaseous stream, which clearly shows that there is a lot of room for enhancement of productivities and selectivities in this particular case. So another quick conclusion is there is room to enhance selectivity towards C3 and C4 with help of catalysts and operation conditions. This is the final slide. I just want to keep you aware that you should always perform the environmental balances and analyze the environmental impacts very carefully, because it's not obvious that something is good for the environment just because it's based on renewable resources. We have very frequently observed in our group that addition of catalysts and use of bioresources can very frequently make the process worse for nature and for the environment. So keep that in mind. We are showing here in all these cases that the addition of catalysts can be very prejudicial, harmful for the environment instead of our general opinion. So finally, the main challenge for sustainable circular chemistry is keeping the carbon circulating in the chemical chain. Plastic provide many advantageous characteristics for development of circular technologies. Making use of plastics for manufacture of LPG through chemical recycling techniques seems to be feasible, technically adequate, and to make sense, and that some technologies, modern technologies that are being developed nowadays can increase both the amount of gas and the C3 and C4 contents of these gases stream. I thank you very much, Priscilla. Sorry.
I will not stay here and wait for questions. I will just...
Sorry for that. Thank you very much, Professor José. So we talked about raw material, how we can convert it, how we can process it, and for different sources and for different ways. Now, to end the cycle, I would like to invite to the stage Professor André Young, that told us how we can modeling and simulate the process that we are developing for biofuels, to produce biofuels.
Good morning, everyone. Thank you. Thank you, Priscilla, for the invitation. Congratulate the organizers for this marvelous event. It's been very good to be here. I think I'll be the only one talking about modeling today, so I promise that I try to not be too much technical here. But I'll talk about modeling and simulation in the production of renewable gases. My name is André Young. I'm professor at UFF. Let me introduce to you the Federal Fluminense University, to those who don't know. We are based on Niterói, in the other side of the Guanabara Bay. If you take the boat, you see our campus in the side of the bay. It's one of the biggest universities in Brazil in terms of students, the number of students. And it has a great difference that we do not have a single campus inserted in the city, but we have several campi spread into the city. It has the advantage to be much more integrated with the society around us. So, and in my opinion, these are the most beautiful campi in Brazil. I'm part of the Chemical Process Integration Group. We are not a group only in Federal Fluminense University, but we also have professors in Federal University of Rio de Janeiro. And our mission is to contribute to chemical process sustainability through research activity in bioproducts, applied thermodynamics, and process systems engineering. It seems to be not connected, but it is very connected, because what we study in thermodynamics is applied in bioproducts, and it's validated, it supports the studies in process systems engineering. That's what I'm talking about here. We have a large group with 6 professors, 2 postdocs, 5 doctorate students, 5 master students, more than 40 undergraduate students. So it's a big group. This photo is from our last barbecue, which was before the pandemic. And this is our infrastructure in Federal University of Rio de Janeiro. We are also part of InSilicoNet. InSilicoNet is an initiative, a consortium between professors and companies in the state of Rio de Janeiro. We are starting the activities. We have started 2 months ago. We already have our website online, so you can find it there. And the idea is to congregate knowledge in several areas of process systems engineering. So you can find partnership in several areas, and the idea is not to only promote projects with the industry, but also to offer courses, to offer tools, softwares for the industry in these several areas. And we already have a short history with Supergás Brás in this project that I think is self-explanatory, technical and scientific elucidation of the influence of ethane in mixtures with LPG, where we are analyzing the influence of ethane in the gas and the tanks. But not only this, I think we are understanding how the composition of LPG, I'm talking about conventional LPG. How the composition influence the properties of LPG. It's conventional LPG, but it will be very useful to bio-LPG, because when I talk to the influence of the composition of LPG will change if we change the raw materials used. So the knowledge we are building here will be very useful to study biofuels in the future. Well, let's talk about process simulation. There are several process simulators available in the universities in Brazil, in other places. Each one of them with their own tools, with their advantages, disadvantages. Some of them are free, some of them are commercial. When you invest on us in our projects, you help us to renew our licenses to those software, and with these licenses, we can teach our students in those softwares, and they can carry this knowledge to the companies where they will act in the future. Why do we perform process simulations? Because we want to save time and money when analyzing process alternatives. To perform an technical economic sustainability analysis, we want to solve mass and energy balances, and we want to solve it rigorously. For this, we need thermodynamic models, equipment models, and efficient numerical methods. If we feed these models with some inputs, we get outputs, and with those outputs, we can get equipment sizing, capital expenditures, adding information about material costs, energy costs. We can perform operational expenditures and profitability analysis. Knowing about the market where we are inserted, we can also evaluate a cash flow analysis, for example, and putting uncertainty in those models, we can perform risk analysis. Knowing about the environment, Professor Zé Carlos mentioned it here, we can perform sustainability analysis, which is very important too. And of course, we can optimize our systems, our processes regarding all these instances. What could be the GIPQ contribution to eventual partnerships? When you have a new technology, for example, a new catalyst or a new adsorbent material, and you want to know how it will work industrially, or you have a process and you want to optimize this process or intensify these processes, we can work together and we can provide estimation of the technology's industrial cost, the impact of the products, the impact of the technology on the product cost, the impact of the product on the product's final price, the economic viability of this technology, and environmental impact of this technology. That's what we do. I would like to show you some experience in biofuels of the last 5 years. We have several papers published in many important journals in the energy and chemical engineering sectors regarding biodiesel, many things about biodiesel, but also biogas, ethanol, we have gasification. We have also some works on green diesel, biojet fuel, from which we can take bio-LPG as a co-product, as was told here. But I want to focus on this one, this paper that's being published right now. It is process simulation of a methanol, acetic acid, and DME integrated production via sugarcane bagasse gasification. The idea here was to propose a flow sheet which has two models. The first one is the gasification, already commented here. The second one is the production of the chemicals. There we can get methanol, we can get acetic acid, we can get DME, and we can get DME indirectly through methanol or directly through from the syngas. It was evaluated economically, and we showed that we can have a multi-purpose plant for this. It's an interesting problem because if we size the equipment and keep the sizes fixed, it becomes a linear problem. So the optimization of this second model will always lead to one or another product. And the best choice would be the DME, direct synthesis of DME, as was presented yesterday. But now let's focus on this first model, the gasification. Because from gasification we take syngas. Gasification has several things that we have to pay attention when we want to simulate it. The first thing is about the biomass. How to characterize biomass in the simulator. It's not easy. And we have several options to characterize biomass. It depends on what we want to get from biomass and what information we have to model this process. We can characterize it by its chemical components, but it's not so simple. For gasification, what we can do is to take it as a non-conventional component and characterize it in terms of proximate analysis, ultimate analysis, and sulfur analysis. In this case, it was sugarcane bagasse. What we need to perform thermodynamic calculations is to characterize the biomass, and you can have this experimentally, in terms of moisture, fixed carbon, volatile material, and ashes. This is proximate analysis. In the ultimate analysis, we quantify the carbon content, hydrogen, nitrogen, chlorine, sulfur, and oxygen content. And in the sulfur analysis, we say in what form the sulfur would be in the biomass. Of course, it's a simplification. And of course, as a simplified methodology, we need to validate it. So when we propose this methodology, we validate it against experimental data. Once the biomass is characterized, it can be gasified. But the gasifier can be a fixed bed, a fluidized bed, or a circulating bed. How to simulate it? In this case, we want to reproduce a circulating bed. It's not simple to do this in a process simulator. What we have at our disposal? We propose to emulate the gasifier as 3 reaction blocks. The first block is a yield reactor where we convert the non-conventional biomass to conventional chemical elements. Then in the other 2 reactors, which are Gibbs reactor, we let the thermodynamic calculate the equilibrium. In the first reactor, we only indicate the possible products. In the second reactor, we restrict equilibrium to what products we want, and we restrict the stoichiometry of the reactor, the reactions, and we let the simulator calculate. We have a degree of freedom, which is the approach temperature, so we can adjust this parameter to reach the experimental results, and then we validate the methodology, and we perform it not only for sugarcane bagasse, but also for wood biomass, and so we validated this methodology, but it will require validation for whichever biomass you have. Once you finish the gasification, you got syngas, and the syngas needs validation against experimental data. And once validated, we can perform sensitivity analysis, and that's why we are performing simulations in the first place. There we can get how the composition of this syngas varies with different temperatures, how it varies with the steam to biomass ratio in the gasifier, and how the syngas yield varies with those variables. And then we can analyze several scenarios in a single simulation, again, saving money and time. It's very important because the composition of syngas will be different depending on which product you want to take from the syngas. This work was concluded, it's being published. I just want to finalize talking about 2 final graduation products that it's under development. The first one is LPG, bio-LPG production from sugarcane bagasse. If we already have the gasification of sugarcane bagasse simulated, now we can produce bio-LPG. How we can do that? We can do by Fischer-Tropsch, we can do by direct synthesis passing through methanol and DME, but in a single vessel. we can do by semi-indirect synthesis, stopping in methanol and DME, and then proceeding to bio-LPG, or indirect synthesis, stopping in methanol and DME, then proceeding to olefins, and proceed to bio-LPG, which will vary. Between those alternatives is the number of vessels and intermediate separation processes. I don't have the results to now to show you, but we are analyzing those different alternatives, and we can do it in a process simulation. And the other work that I want to show you is the work from Ms. Thalita Pereira, who's there, that is under development. The idea is we have a continental country in Brazil, and it was said here yesterday, today, the variety of biomasses that we have and the importance to work especially with residual biomasses. Then the idea is we already have something constructed with sugarcane, but we can also use, for example, coffee or corn, which are very important cultures for Brazil too. And they are, especially in the center-south region of Brazil. And why those three cultures? Because if we take the harvest seasons of those three cultures, they are almost complementary. So if you think in a multi-purpose plant who could process all those three cultures, you could operate the entire year. Yeah. Well, but I'm not processing all the sugarcane. We are processing bagasse, as we are processing, for example, the husks, coffee husks, and corn stover. Of course, it's a matter of logistics to take those residues and take to a plant, not forgetting the straw from sugarcane, just because bagasse is more at hand. But if you can locate some that plant in somewhere, we can take it, and for example, it can be situated in the northwest of São Paulo state, where we have all those three cultures. How do we simulate those materials? It's easy. If they are characterized in terms of proximate analysis, ultimate analysis, and sulfur analysis, we can put it in the simulator, and once validated the methodology, we can foresee what would be the syngas from those raw materials, and then the bio-LPG from those raw materials. And then we want to evaluate it economically, technically, and of course it will require experimental validation in a second moment. Okay? This is it. It's my contact, and I'm free to answer your questions. Thank you.
So everyone, any questions? No? So André, thank you so much. I think this was the best way to see how we can integrate different universities with different raw materials. I'm just figuring out here about the Samba projects that probably we will have more people in the Samba now. Thank you so much for that. And guys, we will have a break now for 30 minutes, okay? And after we will come back for the other part of this first session. Thank you. Could you hear us? Hello, hello. Hello, hello. Could you hear us? Hello, hello. Hello? Eu acho que tá. Hello? Ai. Tem que pessoas têm que saber. Do nada, tô sabendo. Hello?
Hello?
Hi.
Oh, hi there. Can you hear me?
I can hear you. Could you hear me?
Perfectly. Good.
Ah, great. Okay. Thank you, Andrew. We are just waiting for the participants. They will return at 10 in 10 minutes. I mean.
No
Okay.
worries.
And also, if you are ready to share your screen with your presentation, I think it's better to keep it in the screen.
Okay.
Could you share it?
1 moment. Is that okay?
Yeah, that's right. We are see your screen and also hear you. Everything is ready. We are just waiting for 9 minutes. Okay.
No worries.
Okay. Thank you, Andrew.
Bye.
I say, Andrew, are you there? Have we got Andrew online? So change the program around a little bit, and Andrew Welf from the University of Manchester will be the next speaker. Do we have Andrew on audio? Yeah, so Andrew, can we just do a sound check? Christina, have we got Andrew ready to be on audio? We're doing Andrew Welf first, so we've swapped the program around. Hello, Andrew. Can you hear me, Andrew? Hello, Manchester. Yeah. Could we have your points, please, Manchester? I think the presentation will be given in mime. Can you hear me, Andrew? No.
Hello, can you hear me?
Oh, hello, Andrew. Hello, Manchester. Can we have your points, please?
Hi. Hi, Leo.
Okay, thank you. So, gives me great pleasure to introduce Dr. Andrew Welf from the University of Manchester. We've changed the program around slightly, but he will be speaking next on a framework for maximising the benefits of large-scale bioenergy projects. Over to you, Andrew.
Thanks, Keith. Thanks for the introductions. My name is Andrew Welfel. I'm from the University of Manchester, and I work on the UK Supergen Bioenergy Hub Research Network. And I'm going to talk about a framework for maximising the benefits of large-scale bioenergy projects. I'm going to do that basically talking through our experiences in the UK of working on lots of bioenergy projects and basically present our approach for identifying biomass resource opportunities, assessing the economic viability of different bioenergy schemes, assessing GHG performances to ensure those genuine reductions in emissions are achieved, and also finishing looking at sustainability of bioenergy schemes, all important things to make your bioenergy project a success. So if I start from the top and I focus on resources and how to basically identify the resource opportunities, we do this a lot in Manchester and in the UK by biomass resource modelling. And why do we do that? So we do that to basically assess the resource availability in different geographies, whether that's in a country or a region, or wherever you are based in the world. You can assess what will be resource opportunities there. We've done them in all sorts of countries, from Brazil to the UK to Saudi Arabia. Lots of resources everywhere, potentially for bioenergy projects. And this allows you to find the optimal siting for bioenergy plants and for infrastructure, and making sure that you're making the most of what resources are most available on your doorstep. You can also, this resource modeling also allows you to look at the bioenergy potential you generated, and also the biofuel production potential generated. So available, based on what resources you have available, how much fuel or how much energy can you generate, and what can you do with it? Dig a bit deeper, you can look at supply chain analysis. Once you know where the resources are and where your key nodes are, what you would like to do, you can start to work out and calculate how to get things from A to B, and how to do that in the most economically and environmentally friendly way. At a macro level, you can also do supply-demand analysis. So if you're looking at countries such as Brazil or the UK, how much resource is there, how much supply is required and how much demand is required, and balancing those against each other. And I've got some diagrams here on the screen which may be of interest. For example, on the right, you can see this is for a particular map where we're looking at sugar beets in the UK. You can do resource modelling to really identify where the resources are, and that really comes to life in the next diagram, which is more on the forefront, looking at supply chains and working out how to get resources from A to B to where your site is, or where to build your site to start with, based on that availability of resource, and how to connect it to existing infrastructure, etc. So how do you do resource modeling? It's very much match up between these here. So it's looking at food, it's looking at land, and it's looking at what the competing uses are of resources. And I thought I'd share this diagram here, which is basically an architecture of a model we developed in Manchester which we use to basically assess the resource availability. And it looks complicated, but I'll talk you through it very quickly. So in stage one, it's all about assessing the land. So what area of land is built up? What are your forests and your plantations? What kind of agriculture do you have, and what kind of pasture agriculture, arable agriculture? And these are quite important to get an idea of what is going on the land. And by knowing how these are currently and how they may change in the future, you can start to do some calculations of how much land may be available for growing things such as energy crops or biomass crops, whatever that may be. So that's the most important step when you're assessing resource availability. And also building on for each of these, you can kind of build an itinerary of what resources are available, for example, from built up lands. You can calculate the amount of residues generated from roads, municipal areas, etc. Forests and plantations, you can work out how many residues are produced, how much industry takes wood from land, where does that wood go to. You can start to develop an itinerary of what kind of productivity is available from forests. On this side, agriculture is really king in many of the assessments we do, as in we have lots of animals that produce wastes, and also agricultural crops. For every ton of food crop you eat, there's more or less probably another ton of residue that's not edible and may have no other uses. So it's basically doing a calculation of how much resources available. And in the middle here, this is really important. This is the engine, this is industry. And this industry is really important when you do these assessments because they're both a major competitor for this resource with the bioenergy sector, but also can be a major contributor of resources that may be available, again, for producing fuels or energy. Once you've got an idea of how much resources are available, you can start to do more analysis here in stage 3. What technologies are available? How can I convert the available resources into power, heat, transport fuels, or whatever energy vector you would like? And then you can do an assessment of, okay, what do we have at home? Can we meet our targets based on what we have at home? Or do we have to import resources? Or do we have a surplus? Do we export resources? So that's how you assess the resource availability. This is almost one we did earlier. We did this about a few years ago for Brazil, and you won't be surprised, many of you with local knowledge there identify lots of opportunities and opportunity for increasing the availability of energy crop production, for example, sugarcane. This is over the years, and interpreting those results in an energy format. This was our assessment. We did this assessment in 2016, and we did an assessment, okay, where's Brazil's primary energy demand going to be? So this red here demonstrates what we thought the energy situation would be like in Brazil in terms of what you need. This blue dotted line was calculating, okay, if we calculated how much resource we think is available. If you, in theory, were to convert that to energy, what could we do? And as you can see here, this crossover point, in theory, we thought, okay, Brazil could go 100% bioenergy if it so wanted. It's not going to do that because you've got an energy mix and lots of other technologies on the table. But what this showed is a massive abundance of potential resources in a country such as Brazil. And these different scenarios here demonstrate more feasible scenarios of what Brazil could use at home to generate bioenergy from its available resources. And obviously the gap between this dotted line and these demonstrates a potential surplus that could be available for trade in international markets. Okay, so that's resources. Now we're going on to the economic viability of bioenergy. Really, really important. We do techno-economic assessments. Basically, they're quick and cost-effective method to test plans, to identify whether the process is commercially viable or not, to identify key areas for development, and to identify of many options, which one could be the best potentially, from a range of options, which could be the best in terms of feedbacks, technology, crossovers, and what could be most viable. And essentially, it's all about getting you from up this scale here, from a lab level to the commercial level. A techno-economic assessment where each level will allow you to assess whether you think it's viable or not, as if you don't want to build a commercial plant without some kind of modeling beforehand to know whether it's actually going to give you the revenues you wanted. So techno-economics is really important as a further tool in your toolbox to allow you to assess whether projects are viable ahead of putting spades in the ground and building things. How do you do techno-economics? It's very much a case of mapping what's going on in a process, what kind of energy is required, what materials are required, how much do they cost, what's the revenues, etc., etc. And these complicated calculations essentially allowing you to calculate a mass and energy balances at each stage. One of your key outputs is the capital cost, or the capex. How much does it cost to build the initial plant to start with? What are your ongoing operational costs? Profitability is really important, allowing you to calculate things such as the net present value, the payback period, internal rates of return, etc. And you can also take these a bit further by doing a sensitivity analysis, working with different partners to ensure that what you modeled actually relates to real life and what people have experienced. These next tables here are a demonstration of that. These are actually from my colleague Katie Chong at Aston University, where she did a biorefinery techno-economic assessment, basically looking at liquid biofuels from waste, municipal solid waste in the UK. The take-home messages here is, an example, high capital costs to set this plant up, relatively, comparatively low production costs, but each year the revenue would be here. And the kind of the outputs you're really interested in is this particular project had about 14-year payback period. So you have to wait 14 years before you get money on your investments. So these sorts of assessments are really important. They're never perfect, but they provide you a good indication of whether I should go ahead with your project or not, or which decision I should make to go in one direction or another. The next one is really important, which is GHG performances to ensure that genuine emission reductions are achieved. As in, when we're doing bioenergy, and indeed any renewable technology, what's the point of it if it's not going to deliver a low carbon fuel or energy option compared to the alternatives it's replacing? This diagram here kind of places the context. These are our emissions in the UK in 1990. And this is 2018, almost 30 years later. You see a breakdown by different industries there. You can see that we have cut about a third of our emissions, but we and many other countries have ambitions to be net zero by 2050. That's another 30 years from now, give or take. And we've done all the easy stuff. We've got a long way to go. But biomass and biomass sectors and bioenergy are really well placed to contribute to many of these different sectors here. And if it is going to contribute, it's really important that we make sure that it does. Our bioenergy schemes do generate genuine GHG reductions, and LCA provides a tool, or Lifecycle provides a tool to allow us to assess that and make sure that actually happens. So LCAs allow you to benchmark GHG emissions and actually measure the contribution of how a different project may contribute to targets. You can also identify emissions linked to specific processes and activities. So if you analyze a whole process, you can highlight where the pinch points are, where particularly high or low emissions are, and these are processes you could either replicate or you need to really focus R&D to reduce the emissions of a process. It also allows you, if you're doing larger macro assessments, to attribute emissions to different countries and GHG inventories. And this is particularly important if you've got long supply chains and you're moving resources and biomass from one location to another, or you're putting fuels across different international boundaries. The GHG balances can be really complicated, but LCA allows you to work out what's being emitted and you can attribute it to different inventories so it's accounted properly. How do you do LCA analysis? There's all sorts of tools out there. In Manchester, we use one called Gabi. We use SEMA Pro, and we also develop a lot of bespoke models to assess different schemes. This diagram here provides a simple value chain of a given bioenergy project, and LCA works by essentially mapping this out and working out at each stage what energy is put into the process and what emissions or what carbon may be released to the atmosphere or may be stored from the atmosphere. So for example, in the beginning here, you've got the growth of a biomass material with lots of uptake and storage of carbon from the atmosphere. There'll be some kind of harvesting process, you're throwing energy at this. There may be movement of this raw material, so energy being expended there. Then you're converting the raw material to a higher value fuel at a central location. Maybe another transport step when more energy is involved. And then finally, at the place of use, converting that energy and releasing carbon back to the atmosphere. There may also be processes beyond that where energy is required to, for example, deal with waste, etc. So LCA basically adds up all the emissions attributed at each stage of here, and also the movement of carbon, allows you to assess whether what you're doing is overall lower carbon and reducing emissions compared to what you're replacing, which in most cases may be a fossil fuel. This diagram here looks complicated, but I'll explain it. Provides an example of the type of results that come out of LCA analysis. I chose this one. It's for Kenya, not for Brazil, but I thought it would be interesting. It basically shows heat generated from biofuels versus comparators. So these first set of results here are biofuels providing heat for domestic cooking, and these sets of results here are providing heat for the industry in Kenya, in this particular case, the tea industry for drying tea leaves. And these here with the whiskers, little dots, are our assessment of different scenarios, what we think the GHG performance would be if we generate heat from this biofuel. And it's important because each of these are below these solid lines. So producing heat for Kenyan households, we think that bioenergy and biofuels will provide an option to provide heat with a lower carbon intensity than natural gas, lower than traditional charcoal, and lower than traditional firewood. This is even more extreme for industry. So you see the lines here. It's lower than natural gas for heat, lower than diesel, and lower than charcoal. So it allows you to verify that what you're doing with bioenergy provides that low carbon alternative to be viable. Finally, my final theme is sustainability. Sustainability is a really important theme and a buzz theme that comes up again and again, and many people don't understand, well, sustainability is a wild thing. There's all sorts of different issues to think about. And if your project is sustainable, that's great. But if it's not sustainable, I promise you, people will call you up on it and you'll be in trouble in the long run. And bioenergy is particularly important when you're working with Europe, because we have quite stringent, growing sustainability targets with growing stringency. So overarching on an international basis, we've got the SDGs that provide a kind of a framework, and also our climate change targets, Paris Agreement, etc. To achieve these, there's kind of pan-national agreements. In the Europe and still in the UK, we are very much strung to the Renewable Energy Directive. This is the EU policy, and this has all sorts of requirements for bioenergy linked to it. And this is quite important. So if you want to sell your product to Europe and still to the UK, you're going to have to adhere by these minimum requirements. And these are focused around land, as in making sure you're not doing anything with land with high biodiversity value or land that has a high carbon stock. And also emissions for the calculation, making sure your project is sustainable by making sure that the energy or the fuel you're providing has a low carbon performance compared to the comparator. And these requirements filter down at the national level. In the UK, for example, for our fuel, our transport, our electricity, our heat, etc. There's sustainability requirements linked to every level. And again, a step further, at different sectors, sustainability requirements are all over the place, and they're becoming more stringent. And we're developing policy, again, in the UK to take this to the next level, because sustainability does go well beyond just land and emissions, as we'll see in the next slide. This is my opportunity to plug our bioeconomy sustainability indicator model that we've developed in the UK as part of Supergen. You can access it here and read all about it, please do. And we kind of champion the phrase of mapping sustainability. And sustainability is more than just land and carbon. We've identified 126 different issues which we think characterize sustainability. Issues for people, such as health, livelihoods, and society. Issues for development, such as for the economy, technologies, bioeconomy. Issues for natural systems, such as land, air, and water. And then all important climate change, which is very much around the energy system and the emissions associated with those. And we think it's really important to map the sustainability of your projects, because it allows you to develop diagrams like this. It allows you to identify where specifically you can identify the leading benefits. And this is really important for your project and your company, because it allows you to target actions to maximize these and to use them to promote what you're doing. Also allows you to identify where the leading risks are, and again, allows you to target actions to mitigate these risks. I decided to include this diagram here. It looks slightly crazy, but I think it's quite interesting. This is our assessment of linking bioenergy sustainability with the 17 SDGs. And all these crazy links here basically tell a story that we think bioenergy projects, in theory, have a potential link with every single one of the 17 SDGs, which is more so than any other renewable technology. So if you could do bioenergy in a sustainable way, we think you can have a positive impact on every single one of the SDGs, which is absolutely great selling points when you're trying to promote your project. As long as you're not generating impacts, in which case it would be the negative, the opposite of that. So to conclude, some of the themes I've been trying to push today, we think it's really important that you use a framework of models that can be applied, scrutinized, optimized, and make your bioenergy projects better, and also potentially increase the benefits they may deliver. So resource modeling is really important, as it highlights where the leading biomass opportunities are within the geography you're operating, and also allows you to identify optimal sites for building plant and infrastructure. Techno-economic assessments provide that quick and cheap method to test the economic viability of different technology pathways and allows you to inform decisions of whether to proceed or not. LCA are really vital, as it allows you to scrutinize and optimize your projects to assess the GHG performance of what you're doing and ensuring that these genuine emission reductions are achieved. And finally, sustainability is a really big buzzword, and it's really important and mandatory in many effects. And we are really championing a phase of champion the activity of doing full sustainability assessments, as these can provide and identify really clear areas where activity should be targeted, where you can mitigate those risks and maximize those benefits of your projects. And just to finish, it'd be really great. We do all these assessments. We've done lots of experiences doing them. It'd be really great to work with any of you if you would like to do these sorts of assessments on your bioenergy projects. And my colleague Miriam, I think, is in the room. She'd be happy to talk to you, or please email me afterwards, and I will likewise like to talk to you. Thank you for having me today. If you have any questions, please let me know.
I found one that works. Thank you very much, Andrew. Could I ask you to go back to the slide where you had the open access short URL to the sustainability tool? I would encourage everyone just to take a photograph of that. This is, I think, a very powerful tool because, of course, the real challenge that everyone has, especially in Manchester, is garbage in, garbage out is one of the challenges, and it's the accuracy of the data that you have when you're doing these tools. And of course, a lot of the data which is generated can be out of date, or there's a lot of proprietary data, of course. And I think, sort of here in Brazil, you have a better chance of knowing what your own data is. And then using tools such as this cooperation with Manchester to actually, highlight, sort of where it's not black and white, it's always shades of grey, but using these tools I think is very powerful. So, I mean, would you agree with those statements that, it's all about the data that you put in? Because the tools look very powerful.
Absolutely, and they are, all these models tell half the story. You always need to verify as well on projects, and the best models are the ones that use actual data from projects. So 100% agree.
Any questions from the audience here or online? No questions online? Anything from the audience? So you'll be pleased to know that Miriam has made herself known and is busy looking for collaboration opportunities. So I'd like to thank, if there's no questions, then I'd like to thank Andrew in the traditional way. Thank you very much, Andrew.
Thank you, everyone.
Okay, I'd now like to welcome up onto the stage Onosal Yakubulu, who is the lead chemical engineer for SH Energy, and he's got a very important challenge, and that's taking all everything that comes out of the labs and the chemists' view of the world, and then telling us when we're doing it all wrong or whether it will work or not, but also doing those calculations and scaling work from the laboratory to industrial reality. So a very important activity, and one I'm looking forward to him being very successful in. Onosal.
Thank you very much, Keith. Thanks once again, Priscilla, for the nice organization. So I need to push. Okay. So maybe I should go there. I don't know how many of you have watched this TV series Breaking Bad. It was aired, I think, almost 10 years ago. And I think it was in season 3, Walter White, the main character, who later on broke bad terribly. I think there was this episode where he was saying, chemistry is magic. So it really is kind of magic in a sense that, you have the ability to make anything out of anything. So humanity knows, conservation of mass for almost like 400 years now, maybe 300 years. So, if the elements are stable, we can just convert any element to any other formulation in the form of chemicals, unless there's a radiation. And the chemical industry has actually, more than a century experience for these conversion methods. And it can convert a variety of feedstocks into a variety of products using a variety of unit operations. You already heard the biochemical processes, Kaizen or Lanzatec. You heard thermochemical processes like gasification or catalyst, Katya, for example. And the batch processes that we spoke a bit, most of the pharmaceutical industry's batch processes. Then you have continuous processes where you really want to actually go into. So you have a smooth operation, for the same size, you have much more throughput and so forth. Then you have the catalytic process, for example, Keith mentioned already CATIA. Then you have non-catalytic process, which is basically, for example, cooking or combustion. They are the oldest processes, chemical processes that you have. And even the catalyst can change actually a lot. You can have homogeneous catalysts where the reactions are at the same phase with the catalyst itself. It happens most of the times in, say, in one of the emerging technologies, hydrothermal liquefaction. You can have salts dissolved inside your water and then your organic matter as well. And then heterogeneous catalysts, for example, Project CATIA, or which I will speak a bit further on later on, which most of the industry are relying on. And all these, let's say, industrial processes are kind of have a same trend. So first, the ideas are tested. So you have an idea, you know, there's a very typical science hypothesis and all the way scientific methodology. So you need to kind of test the idea, whether it has some validity on it. So then you have very small scale stuff, microliters, you know, micrograms. Then you test something in the lab scale. Then when you have the potential, when you saw the slide, then you go further on all the way pilot and then industrial, if you have the capabilities, budget, you know, policies and etc. So in that regard, the new bio-LPG production pathways are not different. We will have the same methodology. You know, it can be produced from various ways, which I will show you in the next slide. And the ideas are tested at lab scale, just like our projects, and will be upscaled later on, following, let's say, the guidelines of the scaling up. This is a very nice, actually, schematic of the possible bio-LPG pathways, recently published from Argos white paper. Just as I said, variety of feedstocks into, in that regard, bio-LPG, but most of the times it's not only bio-LPG, it has much different components, like addition to bio-LPG. So you have various of processes, fermentation, you know, biochemical, thermochemical, hydrolysis, gasification, pyrolysis, so temperature, heat-based degradation methods, hydrothermal liquefaction, one of the emerging technologies, anaerobic digestion, again, biochemical. Then you have the intermediates, then you have further processing, you know, which goes to the various unit operations and so forth. So of all the things, I would like to focus more on Fischer-Tropsch synthesis, which was already one of the discussion points, you know, with syngas and then one of the previous presentations as well. And the other is methanol to gasoline process, which already makes bio-LPG fractions in industrial massive scale reactors. And also, you know, we will touch slightly on the ethanol to gasoline because it will be transition in the end where we want to use ethanol. So Fischer-Tropsch process is one of the most interesting processes that the chemical industry is pursuing. It dates back to 1925, almost 100 years old. Two chemists, German, Franz Fischer and Hans Tropsch. Ironically, they didn't get Nobel Prize for it. But the first real industrial scale was during the war, when there was this lack of oil. The German industry started making oil from the coal. So basically gasifying the coal, which you already heard today, in the form of biomass. Then you have the syngas, which is a mixture of hydrogen and carbon monoxide. Then over some very beautiful catalysts, they start basically building chain reactions. Keith also mentioned, the chain growth reactions and the theories. So LPG is obviously a part of it. It's just statistically it happens. However, the problem is people don't like LPG. So historically, even though Fischer-Tropsch is very much developed technology, you have massive scales, no one really optimized the LPG fraction. On the other hand, they optimize how not to have LPG fraction. And this process incorporates mainly iron or cobalt based, depending on the temperature, whether you want to have CO2 or not, whether you want to have, let's say, longer chain or shorter chain and so forth. And two, let's say, most common reactors are multitubular fixed bed or slurry bed. The first one is mainly Shell, the second one is mainly Sasol because they are target for different products. And let's say the two most important plants exist in Qatar. One is Shell, one is Sasol, which we'll see a bit later on. The second most important process for us is the methanol to gasoline process. It dates back to the 1970s. And this is a rather interesting one because actually they were not really looking to make gasoline from methanol. It came out of luck. So they were looking for, I think, oligomerization reactions, but then they realized actually methanol made very interesting, let's say, compounds over zeolites. Then they further, let's say, progressed on it. They tested and so forth, and so basically, it's a two-stage reaction. So they have two reactors using the same type of catalyst. The first one is dehydration, which is DME. We already spoke a lot today. And then later on, DME further goes on isomerization or oligomerization reaction to they increase their chain sizes, and then you have olefins, paraffins, aromatics, and you have further, purification steps and so forth. And basically, some of the paraffins is actually byproduct, which makes maybe 10 to 30%. So it has been separated and it's sold as well. And so it's a zeolite-based catalyst, and this technology uses mainly fixed bed or fluidized bed, and a fixed bed mainly Haldor Topsoe. Also Exxon's initial or Mobil's initial reactors were such. Then they shift to fluidized bed apparently, which is already a very interesting, let's say, reactor technology on its own. And as I said, Exxon already deployed in New Zealand years ago, and the recent, let's say, announcement are that they want to deploy again in Chile. And in 2019, Haldor Topsoe-based Thai-Gas Turkmenistan gas-to-gasoline plant has been commissioned. This is a massive, massive, big-scale unit operation. And you see that, they look very different, but yet they are very much alike. That's the beauty of chemical industry. They can learn a lot from each other because there are a lot of similarities. Both are thermochemical processes, meaning that you have medium to high pressure and several hundreds of degrees, which means you know how to handle, let's say, the reactor designs with respect to mechanical designs of pressures and temperature. They're both catalytic processes, metal-based, zeolite-based, or mix of it, and so forth, which also gives you the knowledge of dealing with catalysts, starting up or reducing the catalyst for activity or regeneration and so forth. And another thing is, once you have this kind of core technologies, you have the option to move a similar scale from 1 carbon alcohol to 2 carbon alcohol. Like you shift from methanol to gasoline to ethanol to gasoline, or then later on, even possibly bio-LPG, that's what we want to do later on. And these catalysts are very nice beings, very sensitive, so you can tune a bit them, and then you can have different chemicals or different molecules. You can optimize the use for, just the size, how you, does the, let's say, tune literally the structure of it and so forth. But in all of them, you have the reactor doing the main thing. So it has, let's say, the heavy duty of the whole process. It's the heart of the process. They're all pro and large commercial scales. Even though that you have lots of common aspects, you need to carefully examine every catalytic behavior on its own. So, and this comes to, the art of upscaling, I say. So on the left side, you see a typical bench scale, plug flow reactor. It's heated jacket, or you can also use it as insulation. It is 1 centimeter of diameter. It looks really neat, but I assure you, people who are doing experiments, after some time, it won't look that neat. And then, so at some point, you want to go all the way up to these massive, massive reactors. These are the Shell gas-to-liquids reactors in Qatar, the Pearl plant. So each reactor is 7 meters, and you have tens of thousands of tubes inside, and 13 meter tangent to tangent length of the tubes, but the total reactor is probably 20 meters. So let's a bit, take a moment to appreciate the beauty of such, let's say, scales. You see one individual, how small the person is. I don't know how you look at it, but when I first moved from academia to industry, so I was walking on a 3.2 diameter length of pipeline. So it's really, really big. I mean, we can just fit all of us in just 2 meters. And initially, I was terrified. It was a really, really terrifying experience. It was massive. Then I realized it's actually beautiful. You have the accumulated effort of humanity, hundreds of years of knowledge, and plus, you have like thousands, maybe millions of hours of engineering hours, including welders, construction workers, just put inside this plant. So this is a massive scale plant, very beautiful, very important, and it's like one of the most important, let's say, investment that ever done in the chemical industry. And even you see, how small the CAT vehicle is compared to the reactors. Or take another moment. This is Exxon's methanol to gasoline fixed spec reactor. This on the right hand, you see the reactor itself. It's just one big vessels with some refractory line. And look how tiny the human being is. He's loading the catalyst. That's obviously their zeolite white. And then, they will have to start it up and test and make the safety issue as well. But the guideline, so it's an art, I would say, but you have some guidelines of it. So it is not really, let's say, doing random and trial and error thing. And initially I told you chemistry is magic, but thermodynamics really doesn't like magic. So this is one of my favorite lines ever in an academic textbook. Maybe chemical engineers or chemists know Prasnis polling the properties of gases and liquids. And in chapter 8, you have this beautiful line, face the facts. You cannot get something from nothing. Do not expect magic from thermodynamics. If you want reliable results, you will need some reliable experimental data. So, even you can have, let's say, equilibrium controlled reactions can be known a priori because you have all the fundamental equations and databases. You cannot size a reactor based on thermodynamics. Take, for example, the previous presentation for the Aspen modeling where, we had seen the gas fire. It was an equilibrium reactor. You cannot model your reactor. So you need real, the time-based behavior of these molecules, which is like we call it kinetics. So you really need data if you want to have reliable sizing. And a chemical reactor is most of the times based on chemical reaction rates. So how, let's say, the components behave throughout the time in time domain, how they change, and in the end, how they end up being the way that you want to be in a specific time frame that you want to have. Yet, this doesn't enter. I have seen reactor size because of the transport phenomena. Say, if you have a very high exothermic reaction, you would like to have your reactor be able to get rid of this heat in the sizing. And then you also have flow regime problems. For example, you might have a flow in the reactor that you don't want to have, so you have to design your reactor as such. In all of the cases, still you need to know the reaction kinetics. There are a lot of ways, you have transition theories or RKM theory. They're like very beautiful theories, but sometimes it's really hard to get these theories. They're extremely fundamental, and your molecules are too big to go really fundamental detailed kinetic modeling. Then you need to do some lumped kinetics, but still, there should be something. And these reactions are not a priori, yet there are some methodologies like DFT, density functional theories over catalyst, or Lennard-Jones potentials to see these behaviors. But believe me, no engineer would love to rely on it without doing proper experiments and seeing how it's happening in a slightly bigger way. So they would like to know the reaction pathway because they will be concerned what in a safety situation, if it stops, if there's a safety issue, what are the intermediates and so forth. So they would like to see the kinetic data. And in these aspects, modeling is very useful. Once you have the kinetics, so you can do process modeling, which you already heard for technical, economic, or having a bigger picture. If your reactor is not complicated, you can have mathematical modeling, so one-dimensional reactor. If it is slightly more, then you have to go CFD, especially if you are really caring for hydrodynamics of your reactor, like fluidized BESS. And the modeling is very important because even if you want to move from bench scale to pilot scale, you always have to take safety into consideration. And in that regard, modeling will help you to eliminate some of the safety factors. Plus, a pilot plant is not a cheap thing. It can cost you millions of euros, and which is not an easily justifiable budget to the board. So you would like to have your pilot really, let's say, solid, sound, and like working and so forth. And in that regard, modeling really helps you out. So you have these, bench scale experiments. You have these, let's say, initial data. Then you go to modeling. You size your reactor, and then the modeling will also give you an indication of what are the reaction conditions like, what temperatures, pressures, and then what is the reaction pathway? Do you have some intermediate that you should be concerned about if something happens, in a safety way or so forth? Then the reaction rates, is it really very exothermic, very fastly or not, or other aspects? And so you need to know if it is catalytic velocity, Euler space velocity, how much catalyst you need to put inside that given volume of reactor. And then, like, is it exothermic or endothermic? Do you need to keep the reactor warm or you need to cool it down? So the modeling will help you out. And as I said, like, even the pilot plant needs safety aspects. So you can get away in a policy basis or legislation basis most of the things in a bench scale, but even when you get to the pilot, the safety will be there. So you have to be aware of the safety concerns. So in the pilot plant considerations, so you have to be really pragmatic. You don't need to demonstrate everything in a pilot plant. You know, for example, if you have a process that makes a mix of organic chemicals, and in the end you want to purify some things, you don't need to demonstrate this purification step, because it's already known and very much established. So you just have to demonstrate what is new about that. You know, is it a specific new reactor you are testing, or is it a new catalyst you are testing, or is it a new condition? So you need to demonstrate this one. For example, this is a typical multitubular fixed bed. You have like lots of tubes inside, you have the shell outside, you have the boiling water reactor, or they call steam rising reactor and so forth. So if you're not a pilot, you don't need to do multitubular. It will cost you a lot. It's also very risky to operate if you don't have the enough operational experience. So what you do is you just have one single reactor. And because if you have the proper gas distribution, you will have the identical race. And then industry knows how to get the heat away or how to keep the reactor at the same temperature. And the pilots are also the best, let's say, opportunities to really scale things further on, because it represents the most, let's say, a suitable or identical data for commercial scales. So you should be able to get as much data as possible from a pilot. So compared to, let's say, commercial scale, the cost of instrumentations, the share of the cost of instrumentations are much higher, because you know, the one temperature indicators cost the same, but you will have to put lots of it in a pilot to get a lot of data. And later on, you know, when you do the pilot experiments, you have to see, like, is my catalyst in the right formulation and shape? Did I have, for example, channeling effect? Then you might have to go all the way back to your catalyst and formulate it another shape. Maybe, you know, you have the monolith shape, but it created a lot of pressure drop, or there was a pre-channeling, and then you had hot spots and so forth. So pilots are really, really important, and it also gives you to go all the way back. And towards a demonstration scale, and it is not really just chemical or process engineering task. It is a combination of hundreds of engineers from various disciplines working, like, in total maybe millions of hours of, you know, work. And this is not only engineers, you have construction workers, welders, and so forth. And you don't really use initially just the knowledge of the bench or pilot scale, but also all chemical industry are being utilized. And it is not a one linear direction. You know, you can always go back, which you always have to do. You know, you do data modeling, upscaling, and going back, lab scale, pilot scale demonstration, and going back later on. I think I'm just right on time.
Thank you very much, Anasel.
Any questions, please?
I think we'll save questions for later, because you know, we're a little bit behind schedule, all my fault. But thank you very much, Anasel. And as to sum up the presentation, in God we trust, everybody else provides data, is the mantra of the chemical engineer. Thank you. So now I'd like to introduce you to someone you may not have come across before. But Dr. Miriam Roda from the Aston University is going to tell you about all the possibilities that you could, you know, cooperate with the Supergen Bioenergy Hub. So thank you very much, Miriam.
Thank you, Keith. If I would have known I'm standing in front of like a big screen, I would just have like thought, like, you know, I'm implementing myself into the presentation. I think it's quite fascinating. I might get a bit irritated as well, but let's try how it goes. Oh, how do I go back? Too quick with a help. How do I? Oh, I didn't even press a button. That's magic. Wow. Okay. Let's see how that presentation goes then. So I'm Miriam Roda. I'm from the Supergen Bioenergy Hub, based at Aston University at the Energy and Bioproducts Research Institute. So that's quite a mouthful of hats. I'm having here. I'm going to tell you first what the Supergen Bioenergy Hub is, because that came up quite a bit, and Andrew has talked about this as well, because we work together in the Supergen Bioenergy Hub. So the Supergen Bioenergy Hub is the research network consortium in the UK working on bioenergy. When we talk about bioenergy, we talk about the full spectrum of bioenergy. So we talk about resources, so what are the feedstocks, energy crops, whatsoever wastes, materials we can use then in the bioenergy pathway. We talk about the processing, the conversion, the pre-treatment. So we had a lot of kind of like engineering discussions here, so that is kind of like the part which is interesting for the engineers. We talk about vectors, and then we talk about vectors, we talk about the full spectrum of different opportunities, what you actually can do with your products, with your different forms of energy carriers, and how they work in the market, how they fit the market, but also what is required, what are demands in the market where bioenergy can come in. And then we have also a group working on systems. And systems is quite an interesting word, because whenever you talk to people about whole systems analysis and looking into systems, everyone will tell something that's very, very differently. When I talk about systems, because I lead the work on systems, I talk about how does bioenergy fit into the wider energy system, what role does it play, and particularly how sustainable is bioenergy. We're not just doing research, we also work a lot with internal and external stakeholders. And if I talk internal stakeholders, we're working also with academic partners who are not directly doing core research in the hub, but doing research relevant for us, or relevant to the bioenergy sector. But we also work a lot with stakeholders outside. So we work a lot with people from the industry, because one reason is, or the main reason is, our research has to be impactful. It has to be relevant for the people who actually need the knowledge, need to transfer the knowledge into practical applications. But we also work a lot with policymakers, because our work can inform policy and help to design and shape policy, as it can help to shape and inform decision-making in industry. And one thing I want to mention here as well is we have a really strong early career network in academia, because it's really important to grow the next generation, not just of academics, but also of engineers. So that just kind of like recaps the things we do in the Supergen Bioenergy Hub. And so to get your attention, I put that up first, because there are at the moment research opportunities. So if you have your phone, get a picture or scan the QR code, because there are these funding out at the moment, and when I now talk a bit through what we do in the Supergen Hub, you might think like, oh, this is something I'm actually interested in and collaborating, and then you already know, okay, there's actually an opportunity, so come and speak to me afterwards. Because it's much easier you find me than I find you, because now you all know me. When we do bioenergy research in the UK, and I have to flag up here, bioenergy is actually the biggest renewable energy sector in the UK, so it's more than solar or wind. We mainly talk about net zero, because the UK obviously has a net zero policy for 2050 to reach net zero. So there's a really, really strong focus on the research we do, how bioenergy can contribute to net zero. So it's not just about emission reductions, but we also talk about negative emissions, not just net negative emissions, but also negative emissions. And you met Andrew, and Andrew does a lot of modeling, and we're trying to understand what models actually can do for bioenergy in our research. Lots of talk about models. My colleague Katie, who is a modeler, not a model, she's a modeler, says all models are wrong, but some are useful. And this is why we have done some work on understanding what models are out there to help to model, understand bioenergy as such, for example, process modeling, or resource modeling, to understand where can we grow what kind of stuff or source it from. But then obviously when we talk about net zero, there are also climate change models, for example, to understand the context of bioenergy within these models. And the challenge there is really to understand what can these models do for the research we try to do and to understand, and what outcome can they deliver. And it's really important that there are very different models answering very different questions, and you have to be very careful when you use these models what questions you actually ask, and if you use the right model to answer your question. Within Supergen, we have the people working on resources, and they do a lot of modeling, and they do a lot of resource modeling. And obviously they do that for the UK because we're interested in bioenergy in the UK. But that doesn't mean these models wouldn't be transferable to other countries. Obviously, you need to get your right data in to bring you the results. But there's a lot of modeling going on the GIS level to understand what can be grown where, and also bringing in layers of limitations. So, for example, farmers' decisions. So, because it's always easy to say, oh, there's a piece of land, and we can just grow something. But if the farmer, for example, has to make that decision to move from, for example, an annual crop to a perennial, that is a massive investment decision. And that needs to be considered when we, for example, model, because we can't just say, oh, there's land, and we can just grow, for example, an energy grass, because if the farmer doesn't see the benefit of that in the long term, that decision will never be made. So that's really important to understand, not the model as such, based on numbers, but really the decision-making behind these models. So that's what we do in resources in the Supergen Bioenergy Hub. Oops, that was too quick again. Here we go. On the conversion and pretreatment side, obviously we want to move from polluting industry to green and clean industry. And we have very clever people working on different aspects around pretreatment and conversion. I'm just too enthusiastic pressing these buttons. So we have colleagues working on ionic liquids. And ionic liquids, I'm not going into the detail because I'm absolutely not an expert on ionic liquids, but I try to explain it from a lay perspective because that's how I understand it. Ionic liquids allow you to split your biomass into celluloses, lignin, hemicelluloses, and some other materials. And the colleagues working on that say that's a really clever way forward because it's an easy way of pretreating your biomass. And they say, and I agree because I've seen the results, you can take any type of biomass and also the really nasty biomass because you can nicely handle any contaminants and still get your lignin or your celluloses or whatever you want to kind of like split your biomass into. And you also can use that to actually recover some of the nasty stuff or the metals in the biomass. So this is some work we do in the Supergen Bioenergy Hub. We also work on fast pyrolysis. So I'm not going into detail because I'm pretty sure there are people in the room who know more about pyrolysis than I do. But obviously that's very interesting because that delivers different products, and especially from a net zero or negative emissions pathway, there are many options because obviously that's also when we think about biochar or creating more char-based products. Did I just, no, that was the right pressing. So then you heard about Jude already, and he's working on hydrothermal liquefaction. So that is another option, for example, to collaborate, where a lot of work has been done within Supergen and with especially colleagues in Aston University. And then we also work on catalysis, and within the hub, we particularly focus on photocatalysis, because again, that is an approach which is fairly cheap and can allow you to produce hydrogen and other value-added products. And this is some work where we actually were approached by the Farmers' Union in the UK, who said, well, we do more and more biogas, but the farming sector also has to become net zero. But in farming, you always have these non-CO2 emissions which come from natural processes, and you can't really do something about it, so you always have some emissions left. So they were interested in saying, well, are there options we actually could do something carbon negative? So what if we actually use pyrolysis to treat the biogas and produce a hydrogen-rich gas and carbon black for the industry? And I suggested it to a few colleagues because, as I said, I'm not a chemical engineer. And I had a few colleagues saying, that's bonkers, no one would do it. Why would you do it? You have the biogas and you can just use your biogas. And I said, like, well, but have you at least considered it? Can't we do kind of like a small feasibility study? So one of my colleagues was like, oh yeah, let's do a feasibility study. And she just kind of like ran a few techno-economic assessments, did some very high-level carbon modeling, and realized, well, if you do it right, and depending on how expensive your process is on the biogas side and your feedstocks, you actually can consider it. Obviously, that is on the modeling level, and it's not done in practice, but it is a pathway to consider, because as we heard in the last couple of days, we have to try everything to reduce emissions and find always new pathways to do something about with biomaterials, and to see what options are out there, not from straight away ruling them out. So doing kind of like these small feasibility studies can be really powerful and give new ideas. And as Keith said earlier, the whole work we're doing here in the last couple of days came from an idea and doing a small study. So this is what we like to do. We always say, like, yes, it's nice to get big money, but sometimes it's really valuable just to have kind of like small pots of money and try something out and see if it's actually worth further investigation. And this is now going actually into a big proposal because we realized, oh, it's worth doing, and let's see what's actually happening if we look into it in more detail. So now I'm moving much more in my comfort zone, because now I'm going to talk about life cycle assessment. This is what I do a lot. And as I said, in the UK, the really strong focus around bioenergy is on negative emissions. So it's about sequestering carbon from the atmosphere, doing something with the biomass and locking the carbon away, or turning it into products. We have done quite some work on different technologies, looking at very straightforward combustion pathways, which are really about carbon capture and storage, but we're also looking into more novel pathways. And some work we have done quite recently was around hydrogen production with carbon capture. And often you get ballpark numbers, so someone does an LCA, and tells you, oh, this is the emissions of doing X, Y, Z, and this might be the carbon you can sequester. We got creative, because I think LCA is art. And you can do more than just getting a single number. It gives you so much beautiful information, but you need to understand the context. So this is some work we have done in hydrogen production, where we wanted to know, how much hydrogen can I produce? How much carbon can I remove by doing that from the atmosphere through the biomass? How much biomass do I need to do that? What is the carbon capture efficiency of that process? And what does it all mean together? So what are the trade-offs? So, and just to give you kind of like a really quick overview, and now I actually can be part of my presentation because I actually can reach this. So hydrogen production, your net emissions from your process per energy in the hydrogen, your net emissions per biomass, your carbon capture efficiency up there. It's all the same process. Different results. What have we done? We have played around with the process. We considered different gasification agents. We considered the carbon capture efficiency and what it did to the system. You see, it's 6 different answers to the same question. And what's really important here is how you communicate your results as a researcher, or how your policy is shaped. So if a policy maker tells me, I want loads of hydrogen, these 2, my net negative emissions are actually quite small. If the policy maker tells me, I want large net negative emissions, look at the hydrogen. So as someone who then does hydrogen, or wants to remove carbon from the atmosphere, what option do you go for? if policy tells you, we incentivize you for removing carbon from the atmosphere, you might go for something like this or this, but you're not getting much hydrogen. At the same time, the policymaker might tell you, but you also need to do a lot of hydrogen. Well, you know, it comes with a penalty. And understanding these trade-offs in the system is really important. And that's why I always say, yes, LCA is actually quite useful, but it has limitations, and you really need to understand what you do and what question you want to answer. Andrew talked about the model, so I skipped that, but the link is there again. And if people want to know more about it, please come and speak to me. Andrew talked about it, so I'm not going into detail. One more thing I want to mention, because I'm not just doing LCA, I'm also a social scientist. And for me, doing research is working with people. Not for people, but working with people. Why is it so important? Well, if I do research, and it's not good for anyone, what is the point? It can be nice and beautiful and tell me something interesting, but it's not relevant for society, for industry, for my neighbor. Why would I do it, right? What's the point? So one project I've been working on for the almost last 10 years is working with rice straw in the Philippines, producing biogas from rice straw. Now you could say like, oh yeah, why not, or why would you? The whole point behind this project was to develop a community-based technology which was developed, where the business models were developed together with the people, with the rice farmers in the Philippines. So we spent a lot of time to understand what business models are actually useful for them, and how can we not 100% put that into practice, because obviously there are always trade-offs, but really understanding what is the demand and the need of the rural communities, and then help them to also find a solution which also helps the industrial partner to do something. So really important, not just to go in there and say like, oh, I have a nice technology, I want to do X, Y, Z, and then it's not useful for anyone. We have seen that enough in developing countries where people came in and thought like, it's a great technology, we just do a technology transfer, and then it just wouldn't fit. So the community engagement is really key to success if you want to have a consumer using your product. You need to understand what they actually need and what they want. In the UK, biomass has a really bad reputation. Even though it's the largest renewable energy, it has a really bad reputation. And we're trying to understand why is that, and how can that actually be addressed through academia and support decision-making and policymakers, because we need bioenergy. We have no choice. It is part of the package to deal with climate change. So we need to understand what the framework around policy decisions actually can tell us, and because the policymakers have a social contract with society. And you need to understand that relationship between policy, industry, or the market, and society. And if, for example, the public goes out and says, like, we hate bioenergy, what they do in the UK, at least the media does, the policymakers go like, whoa, yeah, we don't like bioenergy. It's, you know, we're not getting elected if we have bioenergy. But then academia comes in and needs to tell people, or the policymakers, well, what are actually the benefits? And now I'm getting kicked off the stage. So I better hurry up. I still have a few seconds. So we're also working on that. So we're trying to develop frameworks which help us to address these challenges around perception. And as I said, we work with people. And we're not just working with industry and adults, we also work with kids. So a few years ago, we developed a comic, a graphic novel, to communicate bioenergy to a lay audience. This is nice to translate your research in an easy way. But then we thought, like, well, this is what we think we should do. But what about actually the future generations? So at the moment, we're running a project with kids where we ask them, what should energy look like in the future? And that is really powerful, because that tells us, as academics, well, maybe we need to think about our approaches, and maybe what we think is important is not important for them. So that is, and it's fun, because they draw stuff and we have artists helping us to illustrate it. And that is really important, not to forget who we're actually doing research for. Another thing, I'm really quick now. because I know you kick me off the stage. No, I know I have no time, but I still take it. We also work with equality and diversity aspects. So we have a few collaborations in Brazil, actually, where we discuss with partner universities, so we have these two partners here in Brazil, gender equality issues to enable more diversity in the universities, in the research teams, but also for the research we're doing to make it really relevant for different groups in the society. I have about 20 more slides on collaboration opportunities, so I just do that. And people just need to be really, really quick and come to me afterwards and ask me, because there are opportunities out there. It's not massive pots of money, and someone has to write the application. But there are different opportunities out there, so please come and see me. If you have seen something in my presentation you think, this is really interesting, I want to collaborate with you, or at least try to find some money, please come and talk to me.
Congratulations. Thank you. And so, with no more time for questions, there has a question that's come in online, so maybe if you speak to Priscilla and you can answer it to the online audience. So, thank you very much. So, now, thank you. So I know a lot of people were, underwhelmed by Sean's presentation yesterday. So we thought we'd give him a second chance to talk about the deep science of microbes. So Sean's going to talk about, chemicals from waste gases used in microbiology, and hopefully it will be as stunning as yesterday. Thank you.
Thanks, Keith. Thank you, everyone. And thanks again to the organisers. Honestly, before I go on here, I should mention, it's really stunning to me to come to this event. And, for me to stand up here and present is really easy because I get to speak in English, a language that I just about have mastered. But to hear researchers presenting consistently and clearly in English, which is not their first language, is really a privilege. So thank you all for that. So I'm going to quickly rehash some of the stuff I mentioned yesterday, but then we'll start talking in depth about synthetic biology and its potential to allow us to produce really interesting new molecules using biological processes. As I mentioned yesterday, at LanzaTech, what we have done is take an ancient biochemistry, a biochemistry that is likely to have been one of the first ever biochemical processes on Earth, that today exists in a class of microbes called acetogens. And we have taken that capability, one of those microbes, and we have harnessed the advantages of that microbes. We've industrialized the microbes, as I mentioned yesterday. We've developed a process. We've scaled this thing up. We're producing ethanol. We're actually selling the microbe as a protein supplement to animal feed formulations. It has advantages in terms of its circular economic potential. We now operate a commercial process. We have numerous plants. We have 3 plants actually operating today in China, building ones in Europe, more in China, in India, in Japan, etc. So this has become a commercial biological conversion technology. We take the ethanol, we make products from it, and including jet fuel and various products that we can actually start to buy in the marketplace. So what we've actually done is industrialize a biological process in order to allow us to make sustainable products at enormous scale. And the advantage of biology in this process is biology offers an opportunity to harness an amazing catalyst. There have been lots of talks about catalysts, but biology has an opportunity to be an amazing catalyst. A catalyst that can self-replicate, a catalyst that can repair itself, and a catalyst that can adapt to changing situations. So it's like the perfect catalyst that we can operate in our process in gases that are very chaotic in their content. So this is a gas from, for example, the gasification of municipal solid waste. And what you see is the carbon to hydrogen ratio of that gas changes continuously. but our catalyst can continuously produce ethanol from that changing gas at a selectivity of 95%. No chemical catalyst can achieve that. A chemical catalyst will always produce products with, and the selectivity of those products is a function of the feed gas. Biological catalysts are hardwired to produce certain products regardless of what they're fed. We can deal with constantly changing environments, we can deal with contaminated environments, and we can do so for days, weeks, months at a time. Our longest run in one of our commercial plants is 300 days of continuous ethanol production using a steel mill waste gas. So biological catalysts offer an amazing potential. The other amazing thing about these catalysts is they can be programmed. As I mentioned yesterday, synthetic biology is the science of reprogramming these biological catalysts. Understanding the programming in terms of the DNA of that catalyst and then changing that DNA code to allow that catalyst to perform differently, either to be a more robust entity or to make something else, to make something novel. And that is actually what we've been engaged in LanzaTech now for the last 12 years. Synthetic biology is this science. So synthetic biology is the science of reprogramming or re-engineering biology. And it's thought to be the next Kondratiev wave. Kondratiev waves, for those who aren't familiar, are these technical developments that are linked to economic cycles in human society. And we can trace numerous Kondratiev waves in history from the railroads and the steam engines to steel and electricity, petrochemical production, electronic information. And now the idea is that our ability to reproducibly and confidently accurately reprogram biology could be the next Kondratiev wave, because it allows us to harness these biological catalysts to sustainably produce in huge scale molecules that we need today. At LanzaTech, we've taken this organism. So this is an organism that when we started, no one had ever engineered. There wasn't even a genome sequence for this organism. And so we took this organism, we developed not only a genome sequence, but one of the first things we did was to model the organism, to actually to take all of the chemical reactions in that organism, reduce it to a computer program, and then develop and train that computer program to be predictive of how that organism would perform in a reaction environment. We now have a highly predictive computational model of that organism based on its genome that allows us to not only understand how the organism will perform for ethanol production, but also predict how best to modify that genome to allow that organism to produce something else. This computational biology is extremely powerful in allowing us to be able to give scientists essentially a recipe as to how they're going to modify this organism to produce a new product. We use some AI, but generally we use straightforward biological computation. We've developed a biofoundry. What is a biofoundry? A biofoundry is effectively a suite of machines that in combination automate the growth, the analysis, the modification, and development of our organism. This is not trivial. Our organism is anaerobic. It's it's poisoned by oxygen. And so we have to keep all these machines, we have to keep our organism in an anaerobic chamber. So all the work is done with massive gloves in a big glove box, and we have robots inside the glove box continuously and automatedly modifying this organism to the rate of around 14,000 cycles of organism modification per week. We've developed rapid, high throughput prototyping for the organism. So every time we modify it, we can quickly assess what we've done, what the impact is, what the performance is. And we've done all this together with a range of academic, institutional, but also commercial partners around the world. So what does this mean? What's the outcome of all of this? Well, the outcome of all this is captured on this slide. We have demonstrated, we've reduced to practice the production of over 100 different molecules from gas with this organism. Molecules as diverse as acetic, of glycoxylate or glycolic acids, all the way down to long-chain fatty acids or phenolic compounds, or even rubber-like precursors like isoprene. All of these have been produced by modifying this organism in order to allow us to produce new molecules, new solutions for society from the feedstocks that we can process. Not all of these are available now as fully mature technical processes. In fact, I'm going to give you data about 2 of them that are. So we've actually focused quite heavily on the production of 2 molecules in particular, acetone and isopropanol, separately. So we have an organism that can produce acetone. Separately, we have an organism that can produce isopropanol. And we focused on these because, one, these are molecules that are easily separated by the same downstream processes that separate ethanol, i.e. traditional distillation. So when we talk about being able to put different organisms into our established reactors, very little modification is needed for an organism producing acetone or isopropanol because the same downstream process can be used to recover that molecule. These molecules are also used in materials. They're directly used in materials. Acetone is used to make acrylic glass. Isopropanol, it can be dehydrated to propylene to make polypropylene plastic. So now we have molecules that go directly into materials and displace fossil-derived molecules that today are used in that material production. So we can achieve a greenhouse gas reduction or negative carbon emissions by taking an emission, fixing the carbon into a chemical, and fixing that chemical into molecules like acrylic glass or polypropylene that stay stable and fixed for a very long time. So what about production? We're in our lab, we've hit commercial production weight. We define commercial production as 3 grams per liter per hour. For us, 3 grams per liter per hour is where you need to be to have a commercial process. For these 2 molecules, that's what we've achieved. We've achieved that in our laboratory, continuous production at 3 grams per liter per hour, and we've achieved that at a pilot plant, continuous production at 3 grams per liter per hour. That allows us to now start to think about bringing this molecule or this organism, these organisms that produce either acetone or isopropanol into the marketplace. And so we've started here. We want to go other places. We want to go to other molecules. And we want ultimately to be able to offer our customers a spectrum of different organisms that they could make a decision on to put in their reactors at any given time, depending on economic cycles, depending on commodity cycles. Today, I've just annoyed Priscilla because I've asked her to change all my slides because I wanted to update this information. Today, we just announced that we've been able to directly produce ethylene from CO2 using gas fermentation. This is huge. So now you can go to the world's largest commodity. Ethylene is produced at an enormous 160 million tons a year. And we can already demonstrate, we have proof of concept of producing ethylene directly from CO2 in a continuous gas fermentation process. This is a molecule that has a market size of $125 billion annually. Again, something that Miriam mentioned in her talk, if you want to have impact, we've got to be making molecules that people actually use. And we all use ethylene. Ethylene is in this room, ethylene is everywhere. And so if we can make ethylene from CO2, we truly have an opportunity to sequester vast amounts of carbon, but also make synthetic biology relevant to every person in this world, to democratize the technology. I'll get past that. Boop. So, as I said, one of the models we use in LanzaTech to fund a lot of the work that we do in synthetic biology is form joint development agreements. And over the years, we've had agreements and relationships with groups as diverse as Lululemon for fabrics, with Givaudan for fragrances, and Danone for packaging. These are just some of the companies, and of course BASF for a range of chemicals. These are just some of the companies we've had relationships with. All of them recognizing that gas fermentation offers them the feedstock and potentially cost of production advantages that they need in the products that they want to make sustainably. So, going back to the slide I used yesterday, this all feeds into our ambition to enable every consumer to always have a sustainable carbon option for the products they buy in the shops, to be able to choose where their carbon comes from in future, because we can access feedstocks at a price point that makes sense to make sustainable products that people will actually buy, that actually compete and actually displace large volumes of fossil resources. At LanzaTech, I sincerely hope that we can genuinely play a part in a circular economic system, genuinely offer a path to take very, very large quantities of above ground carbon and bring this, yes, into fuels and offer the opportunity to recycle the carbon from those fuels, but perhaps most excitingly into materials that everyday people can purchase, that everyday people can use, and at the end of their life we can recycle and repurpose and almost reincarnate back into new materials for subsequent use. Thank you very much.
Thanks, Sean. Another excellent talk. Do we have any questions? Priscilla?
Thanks, Sean, again for the great presentation. You showed us, the limited to find a way to scale up some process, right, about the productivity. Is there any rationale also to choose to work in the synthetic biology or in the downstream, for example?
Yeah, it's a really good question. And you know, for us, we always have to think about how compatible a molecule is with the biological process, because that always will speak to the concentration of that product that you can have together with the biology, and so while I talked a lot about the disadvantages of chemical catalysts, one of the big advantages of chemical catalysts is they tend to produce molecules at a good concentration in an organic phase. Biology always produces molecules in an aqueous phase. And so in order to recover those molecules from that aqueous phase, there is huge advantage in having higher concentrations of that molecule. And so you're always wondering what impact that molecule is going to have on the viability of your biology, particularly in our case, because we operate a continuous process. So we want the microbe to not only be alive today, but alive tomorrow, and in order to be to keep making. So we have to. That's one of the biggest things that we focus on. And actually, that's something now that we're trying to get predictive about in our modeling. How can we predict what molecules are going to be most toxic?
Great, thank you.
Any other questions in the audience? So, if not, I'd like to thank Sean for coming, and I'm really looking forward to our collaboration. Because one of the days it won't be ethylene that you'll be announcing here. No pressure there, Sean.
You'll be announcing that.
Yeah. I think we'd get Bram for that one. So, okay, thanks very much, Sean. Okay, so that's the end of this morning's session. So we've shortened the lunch, so we'll be back on time in an hour's time. Is that correct, Priscilla?
Yeah,
yes, so slightly shorter lunch. And again, I'd ask the speakers from this morning and from this afternoon if we could just assemble in front of the checkerboard and we'll take a photograph, and the photographers will be, hopefully they're ready to do so. So thank you very much, and see you all in an hour, and we'll be promised to be on time for the online audience. Thank you. frames do work.
This one is also turned on in case you need it.
Okay, it's now 2 o'clock here in Brazil, and we've got our afternoon session, so I'm delighted to bring everyone back where we will talk about more the opportunities that we have for cooperation. We've heard a lot about technology, feedstocks will be important, but also very important will be the funding opportunities that we can have. So we've got various speakers both nationally and internationally joining us and looking at how we can advance the energy transition. So it's my great pleasure to introduce Francisco de Blanco, who's going to talk about the opportunities for Macaúba and complement the presentation we had this morning. So thank you.
Thank you.
The big green button to advance.
All right, the red one to back. Okay. Well, good afternoon. My name is Francisco de Blanco, and I am Solium co-founder and CEO currently. Well, I'm sorry for bringing this huge information about myself, but I think it was important to clarify and settle the expectations about the theme of this panel. Although it is about agriculture and forestry, those are not my backgrounds, right? I'm pretty much a businessman who decided to invest in that field, so do not expect me to be answer to complex questions about the Macaúba itself, but I'm pretty much here to bring my expertise about the rethinking process, then specific about the agriculture and the forestry side of this business. But just to make a long story short, my background is pretty much developing projects, most of them renewable energy projects, and I've done that since 2010, right? Instead, including some very complex projects of natural gas-fired thermal power plants. But in 2018 was the first time I got in contact with something called biorefinery. I started developing a biorefinery in Paraguay. It was a very ambitious project and a very inspirational one too, right? And I can tell that was something that opened my mind in a way I can explain. It was the first time I realized that, for instance, we could take animal fats and vegetable oils and convert those products into perfect hydrocarbons. And that was kind of, wow, we can decarbonize the fuel, the transportation, and save trillions of dollars by taking advantage of the already in place infrastructure. So that was kind of, whoa, we have to do that. And this kind of, let's say, euphoria I had at that beginning kind of became a nightmare a few months after, because then I found out the main problem of this kind of industry, which is the feedstocks, right? It was the first time in my life I was trying to develop a project, and pretty much what I've done, and my expertise is pretty much to understand how to bring complex supply and value chains from the ground and make them work. That's pretty much what I do. So I know that every chain is as strong as its weakest link, and in that case, a huge link was just missing. We didn't have feedstocks to run that plant. So that was when I decided, and I saw the opportunity and I decided to act. And in 2019, I founded Solium. And Solium was founded with the ambition to produce what I call, for many can be a nightmare, because it's not only feedstock, but sustainable, low CI, certifiable feedstock. So it's not simply feedstock. And that was so challenging that I decided not only do the project, not only do Solium to make Solium feasible, but also I worked in other projects to make them feasible, some scale-up project for an oil forest in Paraguay. We launched the Macaúba portal to highlight the potential of this kind of arrangement. In 2023, we are about to launch the Zero Emission Feedstock Alliance, a platform where we intend to support, to develop, and even finance other projects using native trees like Solium. So pretty much what I want to highlight here is the challenge we had and how we tackle that. All right? This is the situation that scared me most at the moment I started looking for feedstocks. All right? Pretty much, we produced 220 million tons of vegetable oils. Or we are about to produce that in this crop to 2021 and 2022. All right? It's a huge number. Yes, it's a quite volume, but and the other good news there, it's this production just tripled in the last 3 years, 30 years, I'm sorry. So it came from 60 million metric tons in 2000, around in the 90s, and it is reaching now 220 million. But the size of the concentration of this market is obscene. I mean, 91% of all those oils produced, they come from 4 sources. And when we zoom it out, we will see that 68% from just 2 sources, palm and soybean. And to get even worse, those 2 sources were on their way, and I think they are still on their way to be restricted or even banned as feedstock to biofuels in the main consuming markets. So that was not possible to establish a business relying on this kind of product, thinking about the future. So that's where I was at that time, and of course it's very concerning. So I start thinking, why are we here? Why this situation is like that? And pretty much, it's not only this. All commoditized business and every business that has, to produce at a large scale, transport, storage, convert. I mean, they need to make the infrastructure as more efficient as possible. And in this case, it happened to be with palm and soybean, and they are feasible sources of these products. But the sustainability aspect of that is what concerns us. And the other thing that kind of scared me is that situation is not that bad when it comes to food. 55% of the total global human calorie intake depends on 10 types of foods, according to FAO. So we have a whole food system also relying on a few main products. That situation explains why it's so easy to get addicted to this kind of supply chain, and that's why we got that addicted to oil, to coal, to natural gas. And the relieving thing is when we decided we were about to do that and start researching what was the best way to do that, because the only thing we knew is we cannot do the same that has been done for the last centuries, for the last decades, maybe century. That kind of agriculture that lead us to this kind of situation, and to the last situation we saw here, that's not feasible, that's not sustainable. So we decided to look for alternatives, and we decided looking and screening the local biodiversity. And that was extremely relieving when we identify in a few weeks of screening more than 100 species of native oils that were potentially feasible to make that a crop. All right. So among those 100, we identified at least 10 of them with production yields above 2 tons of oil per hectare in that stage they are, before any kind of effort, before any effort to domesticate, to improve genetics. So among these species and these materials, we decided there was our first flagship species, and that was the Macaúba, and I will explain why. But after we decided we would working with this kind of material, we had to face another situation. There are no such a thing as villains in nature. When we go to nature, we see that's all about balance. There is no soybean competing, in the Amazon or things like that. What make those things or those crops kind of not sustainable is the way we plant them, is the way we implant them in the nature. So we had to review everything, and we felt very freedom to do this discussion among us, because there were no efforts before to make a native tree a commercial crop before us. So not at least on that region with this kind of material. So what we made pretty much was, let's make something that works and works to everybody. In a way, we can do that forever, and people will want to scale that up because the benefits are immense, are huge for everybody. So let's think out of the box, and let's see what comes and what is the alternatives we have. So pretty much we decided no monoculture was the first decision, and that was very easy. So we had to add and make this kind of agroforestry, agro-silvopastoral systems, and then those prior experiences were already validated, and we could be very confident that this kind of material was feasible and suitable for this kind of arrangement. Deforestation-free, for sure. We cannot cut down any trees anymore. So here the purpose of this business is much on the contrary. We have to replant, to reforest. We have to have highly productive materials to avoid any kind of ILUC discussion. We cannot simply put something there and displace food production or any kind of way people were making their living. So it has also to bring benefits to the environment, to air, soil, water, biodiversity. It had to have a negative GHG life cycle. I mean, CI was something very important when we were thinking about profiting with these materials in other markets like Europe, United States. It has to have also positive socioeconomic impacts, and this is something we are taking a good care of that too. It should help industries, essential industries, to decarbonize their production systems, and as I said, avoid this food versus energy discussion and move towards a food plus energy discussion when we use this kind of arrangements, and live in that vicious cycle and enter into a virtuous cycle, enabling large-scale production. And the way we saw we could do that is pretty much through technology. Today we have capacity and technology no one had when they start thinking about agriculture, I don't know, hundreds, maybe thousands of years, of course, and it keeps growing, but the system is the same. They are just adding some features here and there, but the system is almost the same as forever. So that was a big challenge, rethinking that to make it feasible. And taking the example of the other business I was used to work with, for example, the oil industry. If we consider a refinery had to live only based on diesel and jet fuel or something like that, it wasn't feasible too. So what makes a refinery a feasible and profitable business is pretty much the light ends, the other products that they have, generate value from them, and that's what pay the bills, because only oil doesn't pay. So bringing something that found this kind of situation in nature, all right, this kind of soil, this is pretty much rock, and we planted that tree. The tree is 2 meters tall nowadays, and it is rustic. It is adapted to that region. It can be used in a large-scale system, and it brings positive impacts to the nature because it has capacity to hold moisture for longer during the dry season. It improves the quality of the soil and everything else. So the other aspect we were searching, it has to have multiple products because nothing can be wasted. The zero waste concept trending to a circular economy in a biorefinery just annexed to this kind of system was another important feature for us to make that viable. And then we studied during a lot of time all those biomasses. Professor Avania demonstrated a few of those researches we have done with them, providing these materials and trying to find alternative applications with more value added, making the system even more profitable. So our star of this whole system is the macaúba. Macaúba is a very rustic tree and a very robust tree too, native from the Brazilian Cerrado, which is a savanna-like biome, what allowed us to think about producing that all over other places in the world like Africa, for example, other places here in South America. It survives wildfires, like, for example, it has a very low water requirement. It's part of local biodiversity. We can extract a lot of benefits from that. It has restorative effects, as I said, keeping the moisture on the soil during the dry periods and then keeping all the biodiversity, the soil biodiversity present. Also highly productive. We can reach, now our business model is to produce around 7 tons of oil per hectare, but we already have species and varieties producing more than 10 tons of oil per hectare in the scale. So this is very promising. We are talking about something that it's at least 15 times more productive than soybean when it comes to oil, for example. So it allows us to produce with a lower land requirement, for example. And it has multiple products which allow us to develop this kind of system, a very circular system where we use a lot of these biomasses to produce fertilizer, to produce energy, to produce other things that come back to the agroforestry system and keep twisting and generating benefits. So the system, the agroforestry into the bioindustry at a large scale. This enables us to think about large-scale system, and it generates sustainable feedstocks for all these kind of industries. We can think about bioenergy, sustainable food, biochemicals, biomaterials. We had some samples of the biochemicals, biomaterials, energy with Professor Avania, but here we also have the sustainable food, which was something she didn't approach, but it's also something very promising. We can produce a lot of food from this system too, only from macaúba, but also from the intercrop productions. And just to bring some figures to highlight how impactful this kind of system is, each 100,000 hectares of our system equals to 1.5 million hectares of soybean to produce oil, and the same 100,000 hectares also equals to 100,000 hectares of eucalyptus for charcoal proposal. And just to bring this figure, 70 million hectares, and these 7 millions is pretty much the size of the low productive land we have in the Brazilian Cerrado, including the grade land. So it is a figure that could be easily used in this kind of arrangement. I'm not saying it will be, but it could be. In this size of area, we could produce almost 13 million barrels per day of vegetable oil. It is higher than the US oil production nowadays, and 4 times the Brazilian production. So this is something relieving when we see something like that. We can do that. It's just we need to find better alternatives and better solutions, and even sequestering between 12 to 20 meters of CO2 equivalent per hectare year in this kind of arrangement. So our message here is nature is abundant and we must pay attention and explore alternatives. And we trust so much what you're doing here. We start producing the feedstocks we'll need when we find out the technological solutions to process it. Thank you very much.
Thank you very much, Francisco. Are there any questions from the audience or online? Anything online? From the audience? Can we get a microphone, or shall I? I'll do the running.
First, congratulations for your presentation. Thank you. Just a quick question. The macaúba is resistant for only on Cerrado, or is it possible to produce macaúba everywhere in Brazil?
No, it is specific from the Cerrado area, but we can see it also in some transition regions, in the transition with the semi-arid and with the Amazonic regions, some areas with the Atlantic forest too, but the best place, the most suitable region is the Cerrado. It is a monsoon tree. It needs some wet and dry seasons to produce.
Are there any other questions for Francisco? Otherwise, I'd like you to thank him again. Thank you very much. So now it gives me great pleasure to introduce Dr. Patricia Abdelnour. Hopefully I've pronounced that correctly. And she's going to talk about Brazilian opportunities of raw materials to produce biofuels. Thank you.
Thank you. First of all, I'd like to thank you, the organizing committee, for the invitation. It's a pleasure to me to be here and have the opportunity to represent Embrapa and Embrapa Agroenergy, so thank you a lot. So today I'm going to talk to you about Brazilian opportunities of raw materials to produce biofuels. I will spend just a few slides saying about Embrapa and Embrapa Agroenergy. I don't know if everyone knows about that. Embrapa is the Brazilian Agriculture Research Corporation. It has R&D centers across the world and abroad too. And we are located in this big point there, green, that is in the middle of Brazil, the Brasília. So it's where Embrapa Agroenergy is located. My name is Jotu. We are one of the 43 centers of Embrapa. We are located in Brasília, as I already told you. And currently we have we adopt a manager of working R&D and also technology transfer. I'm the head of technology transfer there, and I'm also a researcher. So we do have four basically axes that we are basically our researchers. That's biomass, what I'm going to talk to you today here. Industrial biotechnology, renewable chemistry, and renewable materials. I'm going to talk about biomass, but I'm going to talk about biotechnology and also chemical process too. Just coming here for the presentation, I'm sorry if I will repeat what the speakers told before, but Brazil is a reference, as I already know, about the renewables in Brazil, in the world. Today we have around 47% of the matrix, energetic matrix, is about the renewables. Instead, in the world we have 40.1. But which are these renewables? These renewables are, most of them came from sugarcane, as you know, that produce ethanol in Brazil. We do have cars, flax, using ethanol too. But it comes from hydraulic, 11% of the matrix, firewood and charcoal, and other renewables. And non-renewables corresponding to 50% that's from, basically came from petroleum and derivatives. If you go to other renewables, we have a list of biomass that we can find in other biofuels too. But I wanted to highlight here just two of them, the biodiesel and also biogas. Biodiesel is responsible for 21.3 of these renewables, and biogas 1.5, but it's increasing a lot in Brazil, as I'm going to show you later. If you go to agroenergy, what's my field of research? We do have ethanol, biodiesel, biogas, diesel, and others. In this presentation, I'm going to focus on these four main materials. I will start with ethanol and sugarcane. That's very important to Brazil too. As you know, the last field we do have 28.3 billions of liters. 87% came from sugarcane and 30% came from corn. But if you wanted to see that corn is very impressive numbers because they come to be 20% until 2030. We have a lot of drop here because of the frost, the drought, and fires, but this is not a very expressive results too. But what research we are doing in Embrapa, according to the first generation ethanol? We are doing GMO processing of some crops, and this is one crop that we developed, sugarcane variety resistance to insects, pests, and herbicides. This is very good result that we have in a program with Embrapa. And you also have the drought tolerance sugarcane variety. So we are increasing the production of them. This is our variety that we discovered last year. That's called BRS 3280 BTRR. That's a resistance for sugarcane and has increasing to quality in the field. Going to the second generation ethanol that came from biomass, we are working on this area too, and we have a development of enzymes and also cocktails to fragment the pretreatment, to do the pretreatment using the enzymatic way. So we do have enzymatics, bioprospecting fungi, for example, to produce enzyme complex, but we also have immobilized cellulose for hydrolysis of pretreatment. In Embrapa AgroEnergy, we have a collection of more than 10,000 of microorganisms, including fungi, yeast, microalgae, and bacteria that's collected from the biodiversity of Brazil. This is used in different kind of projects too. And as I told you, we work with the production of the first generation ethanol, the second generation ethanol, but also the production of bioproducts. And this is an example of production of two acids. The lactic acid produced by bacteria, and also the xylonic acid produced by yeast that are produced in our laboratory. They are still in scale, in laboratory scale. Moving to oilseeds and biodiesel. Biodiesel is increasing a lot, as you already know in Brazil. In January, we have 2% of biodiesel in 2008. And this is going very quickly improving, and it's estimated to next year, it's around 15%. But we do have several regions producing biodiesel in Brazil. The most is the center and south of Brazil producing, is the most producer of them. And we have different kind of feedstocks of the production of biodiesel in Brazil. The mostly more important is the soybean bean. But what is important here is to work to try to doing diversification of the biomass. And in this case, we do working with palm oil and other oils that I'm going to talk to you here, like macaúba, as Francisco told before. Sorry. And about the oil palm, it's more produced in the north of Brazil, in the regions of Pará and Bahia. And we do have a lot of production there, but we have several problems like farrow yellowing, that is a disease that can take that crop. And we are working in genetic briefing program to improving this and to be more resistant to this disease. Another work that we have, we work with the residues of biodiesel production, the glycerin. So we do have biotransformation using different kind of microorganisms to produce bioproducts with high value added compound, like citric acid and kojic acids, for example. If you go to macaúba, I'm not going to talk too much because Francisco already told you before, but we have some projects that with a social way. So, and we do have two kinds of actives here that's called macaúba pump oil by aqueous methods and obtaining macaúba oil by enzymatic acid. In our laboratory, we work even with chemical and also with enzymatic methods too. The last oilseed that I wanted to talk to you is about canola. Canola is an oilseed that's producing mostly in Brazil, in the south of Brazil. But they are producing a lot of the other countries like Canada, like Australia, China, Europe. And in Brazil, it's more producing in the south of Brazil. What we are doing now is to tropicalization this canola to the center east of Brazil. And we have this project that's called Pro Canola. When you have an agreement with farmers, with small and large farms to producing this crop in the Midwest of Brazil. Moving to the last part of my presentation, I'm going to talk just a little bit about biogas. Biogas is increasing in Brazil. We do have, like today, last year, we have a report that showed that we have 811 industrial biogas plants in Brazil that's producing more than 2.82 billion. They are located more in the center and west of southwest and in the south of Brazil, but it's increasing a lot. The region that more has industrial plants are Minas Gerais, and then following by Paraná. So they are the regions that more has biomass plants. And we do have a project that's also a social project in the region, a small region in Goiás, close to Brasília, that's called Lusiânia. It's a small city. When we take the cattle manure, do the biodigestion process, obtain the biogas, electrical energy, thermal energy, and also biofertilizer. That's a very helps people in that region too. For the last, we have a project we're using green diesel. In fact, we have two projects in that area, one using the oil, palm, and another using bio-oil from bones of birds. And this is also in the laboratory scale. To conclude, Brazil is a very important region and is very interesting in renewable energy. There are a lot of opportunity, but what do we believe is we have to do a diversification of biomasses. This is our technology showcase that we have in our AgroEnergy, so you can see all projects that we have there. It's in our website, so we can see all projects that we do and assets that we have there. So thank you for your attention. I'll be glad to answer your question.
Thank you very much. Does anyone have any questions for Patricia? Are there any questions online? Oh,
I just have a question, Patricia. I'm not sure if you can mention, but about the Embrapa and also international collaboration that you have, because it's interesting because Embrapa is really focused on the different types of raw material, but you also have the process, enzymatic biotechnology process. But how you can collaborate, not only related with biomass, but also bring the expertise that Embrapa have from that for the search that you have with others.
Yeah, we are an Embrapa unit. I don't know if you know about Embrapa with EE. It's the research center for industrial innovation, and it has money to funding these projects. It's a non-refundable money. You can use the 130 from the company, 130 for us, and 130 for Embrapa. And you can use it for the all applications, in this case of biomass or pretreatment or byproducts. It's no matter the area, you can use it for that. And international is always possible too. It's possible with the Brazilian company or international company.
Thank you.
Any other questions from the audience? Well, maybe I'll ask one. So, why has Embrapa, as far as I know, Embrapa's never looked at bio-LPG? Is that correct?
Yeah, it's working okay. We are doing, but we are still in the beginning.
Oh, so there are going to be projects at Embrapa on bio-LPG. That is the correct answer. I think a huge round of applause is required for that. So, all in good time, that's fantastic. So, our next speaker is Dr. Edvaldo Murray from LMBR. Edvaldo, thank you very much. And Edvaldo is going to talk about an integrated sustainability assessment platform to aid biofuels development in Brazil. Thank you very much.
Thanks, Keith. Hello, everyone. Good afternoon. And first of all, I'd like to thank Priscilla and Keith for this special invitation to participate of this amazing event. And it's a great pleasure to be here today. And in my presentation, I will talking about a little about the integrated assessment platform that we are developing in the LNBR and to aid the biofuels development in Brazil. And the LNBR is the National Biorenewables Laboratory that is part of the CNPEM, the Center and Research of Energy and Materials that are located in Campinas. The CNPEM is composed by 4 different national labs: the LNLS, the National Synchrotron Light Laboratory, the LNBio, the National Biosciences National Brazilian Laboratory, LN Nano, the Brazilian Nanotechnology National Laboratory. In CNPEM, we can perform different assessment in multi-scale and multidisciplinary research, since from simulations in quantum mechanics and molecular mechanics, passing to advanced equipment analysis like cryomicroscopy, and scaling up of process. And all of this supported by the Sirius, one of the 4th generation synchrotron lights in the world. CNPEM is located in Campinas, 100 kilometers from São Paulo. In LNBR, we use the biotechnology to address scientific and technological challenges of sustainable economic development. The LNBR are divided in 3 main divisions: the Molecular Biotechnology Division, the Technological Process Division, and the Biorefinery and Natural Resource Division, the division that belongs and coordinates. In LNBR, we believe that the sustainability assessment is a way to promote the research and development of new technologies. In this sense, we are not looking just for, in your assessment, not looking for just for the sustainability pillars, but are considered too the other aspects of the Sustainable Development Goals of United Nations. We believe that to be renewable is not enough. We need to be sustainable. This is where the sustainability assessment plays an important role. For example, integrated the process step in a rolled value chain, quantifying the sustainability impacts of the technology, compare different technological roads or alternatives, identify bottlenecks and opportunities, and one of the most important topics that guiding technology development. For example, in LNBR, we perform a continuous assessment cycle. We have the technological development where the experimental are developed, since from bench to pilot scale, and sustainability group. And we share information where, for example, the experimental data is provided for a sustainability group that provide the results of sustainability assessment to the development group, aiding the decision-making process, consider economic and environmental impacts. If you look inside this assessment framework, we have, for example, the process design, technical economic analysis, environmental assessment, and uncertainty analysis. I will show in more detail this platform. We know that choosing technology growth is a complex problem. As I said before, renewability is not implying sustainability. Thus, in LNBR, we are looking to the concept of life cycle thinking. We are not looking only for the manufacturing process, but we are considering the raw material extraction, the manufacturing, transportation, usage and retail, waste disposal, recycling, and reuse. But if we consider a continental country like Brazil, this is much more complex, mainly because of the regional characteristics of the Brazil. Here, we can see how the platform is constructed. We call it a virtual biorefinery that is composed by different modules. We have modules for agricultural system modeling, for biorefinery design and simulation, for technical economic analysis, for life cycle assessment, crop growth modeling, natural resources use and availability, and biodiversity and ecosystem service assessment. And in an external layer, we have uncertainty analysis and spatial analysis. And as a result, we have the sustainability impact. But we are looking for beyond the life cycle assessment. With your platform, we can evaluate, for example, the natural resource availability as feedstock, land use, and water availability. All with this, with sustainability indicators, and in the integrated assessment of the ecosystem. And we perform in some kind of optimization, for example, the rational use of resources, minimizing the production costs and the ecosystem services used, and maximizing the benefits. All of this supported by georeference data and simulations. Some application examples. We already used this platform to evaluate biofuels like ethanol, first and second generation, the use of new biomass, the integration with other biomass, anaerobic digestion, butanol production, lactic acid and other organic acids, sustainable aviation fuels, green chemistry, and more recently we are looking to hydrogen production, sustainable hydrogen production, and some kind of usage of CO2. Some example of our results or outputs of your platform. For example, we can evaluate the breakdown of cost. In this case, we are looking to straw recovered cost, performance sensitivity analysis to the variation agricultural operation. This work was published in 2019 inside the Sucre project. That was a project that was in collaboration with PNUD for United Nations and JEF. And we can perform technological comparison, for example, the carbon and energy balances in the sustainable aviation fuels production. This work was published in an Applied Energy in collaboration with Embraer and Boeing. We can evaluate to the stage of development of the technology. An example here, we can see the second generation ethanol production cost. And we make a sensitive analysis to the variation of process parameters and cost a long time. Here we have the first integrated first and second generation in a short, medium, and long term. It was published in Biotechnology for Biofuels and Bioproducts in collaboration with Banco Nacional de Desenvolvimento, BNDES. This is one of our newest work that is a follow-up assessment where we evaluated the potential for GHG emission reduction. As a case study here was the LNBR enzymatic cocktail. Here we can see a comparison with the commercial cocktail, two different cocktails from literature, and the development in the LNBR in laboratory and in pilot plant. PPDP is our pilot plant. The work was published 3 weeks ago on bioresource technology. And here, an example of our LSCA expertise. Inside Task 39, in collaboration with International Energy Agency, we perform harmonization of LSA tools. Here, we can see for first generation ethanol sugarcane in an approach of cradle to pump, a comparison of the models. In the beginning, we have make great difference between the models, and in the end of the study, we have a convergence for ethanol production. It's important to note that our model don't have significant difference or don't have difference from the beginning to the end. We also performed some other studies for FAME and HVO in HEFA and second generation ethanol. All of these studies were performed under Task 39 too. And some take-home message. we believe that it's very important to make the comparison of technological roads and not only consider the facility itself, but consider the role of value chain, consider the biomass production, the conversion, and the products distribution. To perform the assessment of stage of development to support the R&D and integrate sustainability assessment looking beyond the production cost. And consider the climate change, is considering the natural resources too. Thank you.
No, that's fine. It's always easier to sort of find time. Are there any questions? Are there any questions online? Oh, I think Priscilla's coming down. No questions there. Do we have any questions from the audience? Maybe I can ask, what's the best way to collaborate with LNBR?
Best way to collaborate with us?
You've got lots of capabilities, lots of equipment.
Yes. The focus of LNBR is to develop, to use biotechnology as to promote the use of the new alternatives. We're looking inside the biotechnology as a way to promote this change of paradigm. If it's possible to produce something from biomass, for example, and use enzymes and microorganisms, it's possible to make some kind of collaboration with us.
And so, is this something that you normally do? So, which are the funding agencies that you work closely with?
Normally, we can use the EMBRA-P, for example, as a way to provide funding for the research with companies, for example. But you also have another kind of collaboration. We already have collaborations under ANP. So are kind of some examples.
And for international collaborators, because we've got 50 to 100 people online.
we have a project that is finished in this year with collaboration with institutions from Brazil and Europe under the A2020.
Are there any other questions? Otherwise...
Yeah, as the next session will be online, and we have some technical issues to connect with Professor Glaucia. Probably have 10 minutes break before to start the two next.
That's a terrible suggestion. You can't leave the room.
No, you can leave and take a coffee and just refresh and come back in 10 minutes.
5 minutes.
Okay. Apresentação maravilhosa. Eu tinha visto quando você mandou o final. Obrigada. Tudo bem, a gente teve que parar, porque está muito adiantado e a professora não entrou. consegue ouvir a gente? Você está no mudo. Ah, agora sim.
Ok, olá, tudo bem?
Tudo bem, e você?
Eu tô sem energia elétrica, tô fazendo o meu celular, viu? Vamos ver se vai dar certo. Tô aqui com uma recarga aqui, uma bateria.
Ai, será que vai rodar uma apresentação?
Quer testar?
Vamos pôr pra gente ver? A gente até deu um breakzinho pra gente poder testar aqui.
Ah, sim. Tá conseguindo ver minha tela?
Ah, a gente tá vendo sua tela, sim. Não, perfeito.
Ok? Deixa eu ver se eu der um play aqui.
Não, tá ótimo, a gente consegue ver, sim.
Ai, que bom.
Então tá bom, Glaucia, a gente então entra daqui 10 minutinhos, tá bom?
Tá.
A gente pode até, se você já quiser ficar conectada,
eu fico
tá ótimo.
aqui, né? Eu fico aguardando aqui, né?
Isso, a gente tá vendo a tela do seu celular, na verdade.
Pronto, né?
Não, agora eu tô vendo só... Agora eu tô vendo você.
Pronto. É que tá o delay, eu espero que dê tudo certo, senão você apresenta, né? Você tem a minha apresentação?
Eu tenho, mas eu não sei muito... apresentar, você diz os slides, né?
Isso.
Não, mas sem problema, vai dar certo sim, porque a gente viu os seus slides. A gente só deu um intervalinho pro pessoal aqui e a gente já volta, e aí você põe na tela, se você já quiser deixar na tela dos seus slides, já fica ótimo. Ou na verdade deixa na sua tela para você se apresentar, né, para as pessoas te verem.
Ah, tá bom. E aí a gente faz isso que eu acabei de fazer aqui, tenta jogar de novo. Tá ótimo.
Perfeito. A gente volta daqui a pouquinho, tá bom? A internet está funcionando bem aqui, a gente está te vendo bem. Até já. A gente vai ficar aqui, tá?
So if people could start making their way back in, and then we'll be starting in 2 minutes. 2 minutes. Okay, we'll be starting in 1 minute, so if people could start making their way in. Do we have Glaucia online? So, welcome back. Thanks for coming back. And hopefully we have Dr. Glaucia Mendes Souza, if I've pronounced that correctly.
Hello, yes.
Hello, thank you. How was my Brazilian pronunciation?
It's Glaucia Souza.
Glaucia Souza. I thought that's what I said, but I will stand corrected. And Glaucia is from Biogen FACEP, and she's going to talk about funding opportunities for development of renewable fuels and international collaboration. So given the nature of our online audience and all the different people that we have, delighted to have you with us. Thank you.
Thank you. Just a minute so I share my screen. Can you see my screen?
Not yet.
Okay, I'm gonna try again. Yes?
Yes.
Okay, good. All right. So thank you for your patience. I'm Glaucia Souza. I am the coordinator of FAPESP Bioenergy Research Program. It's a great pleasure to have the opportunity to share with you some of our initiatives and results. So FAPESP is the State of São Paulo Foundation. It promotes technological and economic development of the state of São Paulo and Brazil through fostering of scientific and technological research. FAPESP has several strategic programs. One of them is in bioenergy. And within these strategic programs, we aim to develop technologies. We gear towards applications. We aim for economic and social development. We foster partnerships with private investments. We have several companies that throughout the years have been funding research in partnership with FAPESP. FAPESP also has programs to develop technology centers. Those are projects that develop over a 10-year time frame with more robust investment. that network many research institutions. We have some examples of these technology centers. We support the hiring of new researchers. This is important to give opportunity for young leaderships, young researchers, very talented. So this is an important area of funding. And I'd like to note that for every real that FAPESP finances in research in a small company, that the company generates 11 times this amount in profit or more investments. I say this because FAPESP also funds research in small businesses, companies that have less than 200 employees, and that this has been a very successful program. It's called PIPE. BioOne was created in 2009. We aim to develop knowledge and technologies in all areas pertaining to bioenergy. We work to integrate the community and support government actions. We also give support to many national and international institutions in the construction of standards and protocols, especially towards sustainable practices. And we also develop roadmaps, identify trends, and try to fund areas that we find important. For the next decade, our goal is to increase biomass yields, to improve energy efficiency and sustainability of bioenergy end use. We have, for instance, a technology center with PSA, Peugeot Citroën. This is one example of end use. We have a small company that developed reforming of ethanol to produce hydrogen. So this is also an important example that we can give here. We also aim to develop platforms, biofuel technologies, conversion platforms, biorefinery strategies, and to accelerate the transition to a bioeconomy. The main message here is that we want to broaden and diversify the bioenergy, the energy matrix of Brazil with diverse sources of feedstocks. So BioOne is organized in 5 divisions. We have the biomass division, where we work with many different sorts of feedstocks. We have here breeding, biotechnology, agronomical aspects. We have the biofuels technologies division. Here we develop first conventional biofuels and also advanced biofuels. We have aviation fuels. We have several initiatives in biogas, biomethane. We have the biorefineries division. Here is where we work polymers. We work towards transforming the petrochemical industry, bringing new frontiers, new products that biomass can generate. We have the end use. Here we have long-range station, for instance. This is a call that is open now to fund research on all aspects of long-range transportation, land, maritime, also sustainable aviation fuels, hydrogen, all aspects that will lead to long-range transportation being more sustainable. And we have sustainability and impacts division. Here we have the more horizontal aspects. We look at social, environmental, and economic impacts of bioenergy. We produced over 2,000 papers. This is a network of more than 400 researchers that are leading 277 projects. I mentioned we fund startups. We have over 200 startups being funded now by BioOne, and lots of fellowships in São Paulo and abroad for students and postdocs. Originally, I mean, in the beginning, there was a lot of effort in improving sugarcane, in developing more efficient routes for the use of sugar, ethanol, products that were traditional in the state of São Paulo, have a very established industry. But in the recent years, we see the community seeking funds for other sources of feedstock. So I'm listing here some of them. A lot of research on waste, a lot of research also on other sources of feedstock, wood feedstocks, and residues from the agricultural and agricultural residues other than from sugarcane bagasse. We have projects now that are developing this biorefinery approach. So here we have pyrolysis, we have carbonation, we have biochemical and chemicals that are derived from the biorefineries. Use of CO2, this is an important recent development. We have many different fuels being produced from biomass, biodiesel, ethanol, butanol, biogas, and so on. More recent, we have this trend of biomethane in the state of São Paulo, where you use vinasse to produce bioethanol, to produce biogas. From biogas, you produce biomethane. From biomethane, there is interest in producing green ammonia and possibly hydrogen. And all of this for several different applications, stationary or in transportation. And of course, we look into policies. We have a recent policy nationwide to reduce the landfills, phase out the landfills. So there is here an opportunity to bring value to waste, especially solid urban waste. So we have now lots of projects being called for in different calls of projects, including with the Secretary of the State of São Paulo that works on infrastructure and the environment. We see an opportunity here. This is a new technological frontier. There is need for basic research, so we are funding that, and also looking for partners to scale up some of the applications and results that we are finding. So we want to move away from this, where we have this open system where fossil fuels is being used for most of our transportation, our cities' energy, and the agricultural use, generating waste and emissions that go into the environment and are a damage to our planet. And we want to develop technologies that can bring this system to closed loops. So we have biomass here in the center, substituting for our fossil resources, and this feeding into biorefineries that will bring new products, fuels, bioelectricity, heat, and materials. Those materials that will feed our cities, our industries, our transportation needs, and recycling of the waste, feeding back into the biorefineries. This example that I gave, for instance, of a biorefinery producing from organic matter, such as vinasse, that was usually dumped into the fields. So I call your attention for the study of the International Energy Agency. This is the net zero pathway. They are proposing pathways to allow for a net zero carbon economy by 2030 and 2050. And if you look at the blue bars here, those are technologies, most of the technologies that are needed for net zero are already in the market, okay? So by 2050, 50% of the technologies that will be used are already in the market. So we want to see what can be done with technologies that are already here, that could be scaled and stimulated. So Brazil, of course, is a reference in this field. We have been able to achieve a largely renewable energy matrix. We have an energy matrix that is derived from sugarcane that contributes significantly to our electric matrix. We have over 1,200 commercially operating mills for ethanol, for biodiesel, for biogas, and for biomethane in Brazil. So there is already an ongoing industry. If you look on the demand side, we have flex fuel engines creating this demand for biofuels in a very successful way for many years now. We are doing all of this using very little land. Only 7% of the land used is for bioenergy, and we have a policy framework that is rewarding the savings in emissions. So all of these have been contributing to a lot of CO2 reductions in the last 20 years. This would be corresponding to 4 billion trees. So Brazil was invited by the Scientific Committee of the Problems of the Environment to lead a report on bioenergy sustainability. SCOPE is an organ that was created by the United Nations to aid the United Nations Council on Problems of the Environment, and they wanted a report on bioenergy. BioN was invited to lead this effort. We produced many documents. I invite you to go to this link. They are all open access. And we concluded, one of the conclusions of this study is that there is 500 to 900 million hectares of land in Latin America and Africa that are rain-fed and that would, if used for bioenergy production, not compete with the forest, with the native vegetation land, or food production. So the International Energy Agency, BioN contributes and leads actually Task 39, which is the task force of IEA Bioenergy for decarbonization of transportation. And Task 39 asked us to conduct a study on emerging economies. So we will launch the study soon, but I can advance to you some of its conclusions. We mapped the Global South, looked at policies. If you look at the countries in blue, in Latin America and Asia, they are the countries where biofuels policies are fully implemented. You have the countries in salmon, in green, where it's partially implemented. That includes India and China and Australia. And we have those countries in salmon where there is an environment, a friendly environment for biofuels deployment in scale in a medium to short term. So we did a life cycle analysis integrated with a techno-economic analysis and an exergy analysis of biofuels in these countries. We started with Brazil, Argentina, Guatemala, and Colombia. We are now doing China, India, Malaysia, Thailand, and South Africa. But what we saw so far is that biofuels are contributing in these countries significantly to reduce emissions. If we rewarded these economies using a low carbon policy framework, we could bring around $630 million to their economies, to these four countries, that they are competitive. So the economics is competitive in relation to fossil fuel prices. And we found points where we could improve their systems doing an exergy analysis. So I conclude by making our program at FAPESP available to foster partnerships and co-fund initiatives that you might find interesting. And we have a very positive view of bioenergy. We are looking into biofuels in several countries, and I think it's very promising. We should continue towards this effort. So I'll stop sharing here my screen.
Thank you very much. And the iPhone is an amazing device, isn't it? So I believe there was a power cut and then you had to sort of revert to your phone. So thank you very much. I'm amazed that worked as well as it did because of doing it on the iPhone. But anyway, so thank you very much. Are there any questions? Maybe just for my benefit, you only fund within the Sao Paulo region, is that correct?
Yes. So by constitution, the state of São Paulo created this foundation. It funds only research led by a researcher that is located in the state of São Paulo, but that does not mean that we do not conduct research that is taking place in other states. We have, for instance, a call for projects on the Amazon. Several researchers in the state of São Paulo collaborating with, of course, researchers from the Amazon region. So it's possible to do research in Antarctica, in the Amazon, in other countries.
So I think the BBSRC in the UK has a research cooperation. And are there others in other countries as well?
Yes, there are many. With Germany, with the Netherlands, with Japan. There was an open call recently with Japan. We, of course, collaborate with IEA. Under IEA, we are collaborating with 14 countries under Task 45 and 16 countries under Task 45 that works on sustainability.
I look forward to finding out all of the countries because we have operations in many of those that you've just mentioned. So thank you very much. And if we could just give a round of applause. Thank you. So I'm now pleased that we have Bruno Bocci from FINEP. Thank you, Bruno. And he's going to talk about funding opportunities for the development of renewable fuels. So thank you very much.
Good afternoon, everybody. I am Bruno Bocchi. I work at FINEP at the Agribusiness and Biofuels and Food Department. I'm going to show a little bit of finance opportunities in Brazil. I want to thank the organization because it's a very good event that was held today. First of all, I have a disclaimer. We are under electoral period, so this presentation is merely informative. It's related to FINEP's mission. Only neutral language is used without any opinion. And so it's not going to be a pitch or a seller presentation today. I'm really sorry. I can't do that. I have to wait 1 month to do so. FINEP is a state company vinculated to Ministry of Science, Technology and Innovations. We are a state company, but we work more like funded agents. That's because we do a lot of bank operations, so we need to be a company to do so. FINAP's mission is to foster innovation in companies and research institutes, startups, all agents in the innovation chain. We work from technology readiness level, from basic research until market introduction, production, pioneer production. So we work all in the innovation chain. We have most available 3 kinds of support. We have other ones, but I'm talking only about that. We have grants, investment, and bank financing. So we have some open calls. Right now we have the biotechnology call with 50 million reais. You can use it to human health, agriculture, environment, and industry, including biofuels and biochemicals. We have also open call for FINAP, we call it FINAP IP. It's for projects between universities and research centers and companies. So it's a continuous flux. Everybody can take this resources until the resources are available. So it's going to, how can I say, extend for a while. You have more than 1 year maybe with these resources. We have also renewable energies call, more with a focus on university and research centers. We have also FINAP Startup. It's a continuous flux operations also. A startup can get up to 1.5 million of investment, and the startup must present revenues from almost 400K up to almost 5 million annual sales. We have also bank finance. That's a shelf product of FINAP. So we can support it with the finance. We can support all kinds of activities, from construction, software team, technology transfers, first production, pioneer production, startup acquisition. So it's a very, how can I say, you can use whatever you want with this money. Sorry, can I do that? How can back here? Go back? Yes. Okay. The projects are evaluated. We're using innovation relevance and innovation levels with all of those aspects here. And with we analyze the innovation level, the relevance level to analyze the project and to look for what is the best condition that the company can have with FINAP. The proposal analysis time is really short. It's only 45 days until you have a decision to support the project, and you take more 30 days to liberation, so you can have a project with less than 90 days. So it's really fast to get resources for with finance. Those are the conditions, direct operations with FINAP. We have today interest rates from 5% up to 6% per year. So it's a competitive rate in Brazil. We are under a provisional measure today. So credit conditions are indexed to a referential rate. Otherwise, different conditions may apply. We think in February you have a final solution for this for that. But right now, it's a really good condition for credit. The months and the total period are really extended, so it's really to foster the innovation in companies. We have some additionals with projects between universities and companies. We have some subjects that are really fostered too, like IoT, sustainability, and education. Well, if you have any doubts about the presentation, I'm really sorry, because today really can't do I can talk much about FINEP or numbers or sales numbers or things like that. It's a really protocolar presentation today. So, but if you have any doubts, you can. I think we have time.
Thank you very much. If we want to thank Bruno for the presentation. But don't go just yet. So I understand you may not be able to answer all questions, but historically, have you, the chance of success in research proposals, what is that for an academic historically? No, the percentage of grant applications that are awarded. So.
It really depends on the open call. I think it's normally really 20%, 10%. It depends on the resources available. Normally, if you have a huge resource available at the call, you have a higher rate. So.
But there's going to be there's probably a forward-looking question. But historically, you funded in biofuels consistently, so we could hope that will continue in the future, but I understand you won't be able to comment on that. Are there any questions in the audience or online? So for international collaborations, so for organisations outside of Brazil, then how have there historically been funding calls for international cooperation as well? Could you comment on how they're structured?
We had some calls available with, no, not now. Now we had in the past. We had with UK. We had also Germany, Spain. I think US. You have. It's a little difficult because you have to combine two agents to settle up the subjects, but you had in the past 3 years you had some calls, but it's not so common. I think it's really difficult to combine the interests of both agents. So I think it's the main problem, and you have to have resources for both sides available. But you had in the past a lot of calls about it. So,
I look forward to more calls being available for international cooperation. We have operations, we have university partners in all the countries that you mentioned, and in the future, then we look forward to working with our network and our ecosystem of researchers and companies so that they can cooperate with Brazil with some public money to help things go along. We do fund, sort of directly, but we always find that working in cooperation with public funding can help accelerate and increase the impact for our research. So thank you very much, and I look forward to you being able to give judgments and opinions and tell us about the future. So thank you very much. So, do we need to do a quick sound check, Jack? Are you good?
Can everyone hear me okay?
Yes, we can. So, if we're all good, I'm not hearing anything negative. We're slightly ahead of schedule, but that's always good at this time. So, Jack Leonard is from the DOE and the ARPA-E program, and he's going to talk about some very interesting projects that they've been doing, and hopefully some opportunities for international collaborations. So thank you very much, Jack.
Okay, and thanks for the invitation. I appreciate you guys squeezing me in at the end here. I guess I will be running the slides from my computer, is that correct?
Let me put you over to Priscilla, because she knows more about such technicalities than me.
Hi, Jack. So if you can share your screen, will be better, mainly to get the control for the slides and avoid any delay.
Okay. And can you let me know, can you see the slides okay?
It's coming. Now that's okay.
Good? Okay. Well, thanks again for the opportunity to talk about some of the activities here at ARPA-E, as well as the broader Department of Energy. I think we all share a mutual interest in a sustainable future. So what I'd like to do is talk about first a quick overview of the U.S. Department of Energy. It's a very large organization, and I represent just a very small group within it. And then talk about what we're doing in renewable fuels and opportunities to participate in an ARPA-E project and with the ARPA-E teams. And then also shift focus to talk about some of the international efforts, particularly with regard to Brazil. So with further ado, let me talk a little bit about the U.S. Department of Energy. This is the org chart that we have for the U.S. DOE, and you can see it's quite complicated. I'm just circling 3 small organizations within this enormous group here. Our own organization, ARPA-E, and then I'll talk a little bit about the Office of International Projects, and they are working with our EE and RE group, the Energy Efficiency Group, which has an office called BETO, Bioenergy Technology Office. And so we'll be talking about that group. Just to give you an idea, ARPA-E, I mean, the Department of Energy has a multi-billion-dollar budget. ARPA-E's budget for next year is about $450 million. So we are a relatively small group, but we're a new group. And interestingly, we report directly to the Secretary of Energy, which reflects the origin of our organization. So maybe I should say a few words about ARPA-E. We were created about 10 or 12 years ago with an unusual mandate. We were told to fund the research that's simply too risky for the private sector. There was a notion that there's a lot of good ideas out there that simply can't get that first dollar of investment. And so we were created specifically to identify disruptive technologies that could have a high impact, but we're simply not getting attention from either other government agencies or from the private sector. And since then, we have launched many, many programs. The way ARPA-E works is that program directors like myself are brought in for term limited periods, typically 3 to 5 years. And so there is no set agenda. Program directors come in and they, I don't want to say get to work on what they want, but we have very broad purview over what we can work on. And in my case, I'm interested in issues around decarbonizing the natural gas grid, reducing methane emissions, but also, and in our open call where people can propose anything as opposed to a structured program, like say, some of the ones that are circled there, in the open, people can propose any ideas. And there we look for really, completely novel areas. And in the context of this meeting and our discussions with SHV, one of the teams that I had known from my time when I was at the Gas Technology Institute is a team from IIT who's been developing electrochemical reduction routes. And when I first talked to them, they were focused on making carbon-carbon bonds as opposed to just reducing CO2 electrochemically and making, say, formic acid or perhaps methanol. And they were quite successful with making C2 and C3 bonds. And when they put in a proposal, they said they wanted to work on ethanol and propanol. And I thought, well, this is a very interesting project. We will fund this. And in talking to them, I mentioned that, propanol is interesting, but I know the propane industry would be very interested in turning that into propane. They said, oh, we can make propane. We just didn't think anybody was interested in propane. To which I said, well, I think there's an entire industry that's quite interested in that topic. So we've since had some opportunities to talk to SHV. And I think that's an example of how we can collaborate, even if it's on an informal basis. Let me go forward here. Hold on a second.
Let's go.
So the way ARPA-E works is we are a funding agency. We actually don't do the research ourselves. And over the past roughly dozen years, we've handed out about $3 billion in funds. We've funded 1,200 projects. And there's a lot of ways to participate in ARPA-E projects. One, you could be part of the team. Two, about 15% of our awardees ultimately get follow-on funding from the private sector. And so you could participate by joining a team or working with a team, even if it's outside the scope of the ARPA-E project, and working with them. And then we've had several teams that have actually matured to exits, and those companies have roughly about $20 billion in exits. And so there's an opportunity to participate as either acquirers or licensors or partners of companies that come out of the ARPA-E work. There are a lot of rules, though, for participating in an ARPA-E project. The U.S. Department of Energy obviously is using U.S. taxpayer dollars. And so there's specific rules about how foreign entity, non-U.S. entities can participate in an ARPA-E project. And I strongly encourage you to follow the link. This is to our frequently asked questions. And particularly in section 2 and 3, a lot of these questions are answered in much more detail than I can go into today. The first thing you need to do, though, to participate in an ARPA-E project is you have to know there's a project. And what I'd encourage you to do is sign up on the newsletter, and then you get the latest funding opportunity announcements, or FOAs. These are essentially our requests for proposals, where we announce that we're going to have funding available in a specific area. So if you're a non-U.S. entity, you are eligible to apply for funding for ARPA-E. However, ARPA-E can only make the awards to fund a U.S. affiliate or a subsidiary entity. So in other words, someone who's incorporated in the U.S. or one of the U.S. territories. Work must generally be done in the U.S., but foreign work waivers can be applied for, and they do require special approval. I will say, in my experience right now, I'm actually funding a team in Australia. They had some very unique capabilities in terms of their modeling work, and so we were able to get a foreign work waiver for them. In another program, I have 2 companies, again, one from Australia, interestingly enough. We're working with their U.S. subsidiary, and another one is a Canadian company. Again, we're working with a U.S. subsidiary that they have. But in both cases, I would say, Australian and Canadian nationals were heavily involved in those 2 programs. So what I'd like to suggest is, if you see areas of interest, please approach us and we can see what is possible. When it comes to follow-on funding, there's basically no restrictions on what you may be able to do. As I mentioned, about 15% of our awardees do attract private funding from outside the government, and that is open to anyone to participate. And what we've seen is a full spectrum of involvement. We get a lot of funding, follow-on funding through venture capital. Sometimes that's in-company venturing arms of the company itself. There's a lot of partnering, licensing, and even acquisitions that goes on. So there's certainly plenty of opportunities to participate. When it comes to international programs, I want to emphasize, I'm not the expert in this area, but we do have our international office. And there's a good news, and maybe for some of the folks here, because I've been listening to the meetings all day long, and maybe a little bit of disappointment for others. So our Office of International Affairs, the way they work is they coordinate international programs with DOE agencies. And I mentioned before the EERE organization, and specifically their BETO, or Biotechnology Office. So the Office of International Affairs doesn't fund the projects. The funding comes from these DOE agencies. And in this case, there is a bilateral framework with Brazil that's been in place. MOUs that were put together almost 20 years ago, and another one back in 2011. And the Office of International Programs is right now working with BETO to kick off a program that's focused on sustainable aviation fuel. And SAF, as you know, is a huge topic. All kinds of routes, everything from vegetable oil hydrogenation, biodiesel, conversions into renewable propane, and of course jet fuel, as well as electrochemical routes and various chemical feedstocks. Even biological routes are being proposed at this point. And I'll mention that I saw the LanzaTech presentation. Ethylene is a great precursor into SAFs. So the BETO organization is just getting this program kicked off, and that is one that's I would say is readily available for Brazilians to participate in, and people can be from the private sector, universities, etc. When it comes to propane, which I know is the reason I got involved in this discussion in the first place, unfortunately, there is no program right now in propane. And when I probed on this, the question would be, if there were to be a program in propane, it would have to be by mutual agreement for both the DOE and the BETO office, most likely, as well as the Brazilian government. So at this point, all I can say on the non-aviation fuel side is, if there is interest in doing something in this area, this is the time to start talking to your officials and see if we can put something together. So I'd like to share some final thoughts, and these I want to emphasize are my own. This is not DOE official policy by any stretch of the imagination. So the first thing I want to point out is, because I know there's a lot of interest in a broad range of fuels, but I'd like to turn the attention back to renewable LPG. And I think renewable LPG can be much, much more than dimethyl ether and propane. I think there's a suite of molecules, or perhaps blends of molecules, that may be of interest here that could essentially act as drop-in fuels. And I would hope that there's opportunities to work with you to identify those. I think the other thing is renewable LPG can address many more markets than today's LPG customers. In addition to going beyond heat and the limited transportation for, say, propane autogas, some of these molecules like DME are a drop-in for diesel. I think there's opportunities to decarbonize the natural gas grid. My former company converted a gas grid that ran on propane to natural gas, and we could just as easily convert one from natural gas to run on renewable propane. It's quite fungible, and I'll add that in the U.S., the natural gas market is probably 20 times larger than the LPG market, for at least for residential and commercial heating. And the last thing I'd like to point out is that there's work in carbon capture, which would be the source of CO2 for many routes to renewable propane. There's a lot going on in hydrogen electrolyzers, which would be an important intermediate that we need if we're going to reduce CO2. I think we heard from Sean, but there's obviously a tremendous amount of work going on in fermentation, in electrochemistry, and in catalysis. And I'll note that congratulations to the Brazilian researchers who have just published some work on a direct route to oxidize methane to methanol. Quite exciting, because then that opens up new routes to DME, which is a great substitute for propane. So all of these efforts are out there, but these efforts, while they form foundational chemistries and processes that could be pertinent for LPG, they're not focused on that at all. So I think that there's opportunities, if you look at some of this work that's going on, parallel efforts, and ask how can this be applied to the LPG world, there's a lot of opportunities to, if you will, steer some of these things through the collaborations into the LPG direction. So that concludes my comments. Again, I very much wish I was in Rio instead of here, but this is the next best thing. So I hope there's some opportunities to keep this dialogue going. And thanks for your attention, and happy to answer any questions that I can.
Thank you very much, Jack. Can we thank Jack?
Obrigado.
And one thing that Jack said was that he will fund projects that won't be funded by other agencies or the private sector. I can fully agree there, because there was the project he mentioned at the IIT, and when I saw that work, I thought, well, no, that will never work. But then the researchers presented the data, and we are following up on that. Is Jack still there with the video? Can we get him on the live feed?
I can figure out.
Oh, you're still there, Jack. Okay, that's good.
Yeah.
Yeah. Did you get a chance to see my presentation yesterday?
No, sorry. Yesterday was a federal holiday, and I was actually traveling, so sorry about that.
Well, if you had, you'd have seen that we've developed a process to convert ethanol into LPG with aromatics, which could, of course, be sustainable aviation fuel, with a 2-to-1 ratio of LPG to sustainable aviation fuel. So we did the first technical reveal of that yesterday. Maybe I'll reach out to you next week when I'm back, and I'll give you that presentation.
I would be very interested. Yeah, and I think others here in the DOE would be as well. This is a very, very hot.
And I think it will be of interest to both the US and the Brazilian governments as well. So, are there any questions for Jack? Priscilla?
Guys, could you come back, Jack, on the screen, please?
Oh, yes. Let me see. I'm trying to figure out how to stop sharing here. Sorry. Give me a second here. It's...
Yeah, and... Podem pôr ele de novo na tela, por favor? So, Jack, recently we were in the GCAF, and we saw that heard that the U.S. government announced a lot of investments, mainly in the hydrogen hub and for a renewable hub, right? Do you think that for these hubs is really specific for this kind of sustainable fuels, or we also have some opportunities to include other sustainable fuels like LPG, RDME, and also collaboration between countries in this hub?
So I know the hubs in general are looking for a diverse set of hydrogen applications. When I've talked to some of the organizations that are contemplating putting in a bid for the hub, they typically are looking at both large-scale applications, hydrogen for, say, transportation, chemical use, industrial decarbonization. But the DOE is strongly encouraging teams to present a whole suite of applications for hydrogen as part of the demonstrations. I'm not aware of anybody who's working directly on DME. I have actually been promoting that concept, and I think others are. I would not be surprised to see some of the hydrogen finding its way into the methanol market, just because that's a major chemical down in the Gulf Coast, and there's chemical buyers who will pay a premium today for renewable methanol. Again, what I would strongly encourage, most of the organizations that are pursuing hub projects are quite vocal about their interests in pursuing projects. Unfortunately, as a federal employee, I can't appear to be encouraging one potential applicant over another or seeming to favor one. But I think if you were to look at who is forming hub projects, I think you would probably find at least a few teams who would be very interested in talking to you about your ideas for using the hydrogen in the renewable propane space.
Thank you. Great.
Are there any more questions for Jack? If not, I'd like to thank you, Jack, for joining us. You missed a great event, so but if we organise it again, I'll give you plenty of notice and look forward to seeing you here in the future. So, once again, if we can just thank Jack. So, before I hand over to Rebecca, who just wants to say a few thank yous, I just want to thank the technical team at the back, the photographers. You've been working, sort of with a hybrid event, which is always the biggest challenge going. So, thank you very much indeed. If we can give the technical team a round of applause. And I'd just like to hand over to Rebecca.
Thanks, Keith, and thanks everybody, because honestly, as we saw yesterday, it's a team sport, this innovation in bioenergy and in solutions that we need for sustainability that moves the world. So indeed, the technical team, the translators, everybody who has supported in organising this, please give yourselves a big round of applause, because we really, really appreciate everybody's attention and detail. But there are two people who I really need to single out, because they have been working round the clock and really put their heart and soul into the organising of this event, and it really means so much to us as SHV Energy and Supergas Brass. So on behalf of both of those organisations, I would like to ask Priscilla and Keith to come up here on the stage. Yeah, go that way. Oops. So, first of all, don't worry. First of all, Keith, Head of Research and Development, Principal Dancer, Organiser extraordinaire, thank you so much for everything you do for SHV Energy, for the renewable liquid gas industry, and also for being a genuinely nice bloke.
It helps when you've got a good boss.
And of course, everybody here in the room and everybody watching knows Priscilla, because she has reached out to everybody personally, has been involved in everything that has been organised over the last couple of days. And it's a really amazing experience to have 2 whole days around bioLPG and renewable liquid fuels. So we're really, really grateful for everything you've done, Priscilla. On behalf of SHV Energy and Supergas Brass, big, big, big thank you. So, thank you, everybody else. And now I'm going to hand over to Rubens, who's also going to give the closing ceremony. Thank you so much.
Thanks, Rebecca. Agenda. I hope that all of you have enjoyed a lot the agenda that we covered in these 2 days. It was very intensive. I think that everybody learned something new. Everybody had a good idea to continue our work. This is just the beginning. Those kind of conversations will be more and more important in order that we achieve the journey that we would like to do in our sustainability. So, on behalf of SHV and Supergas Brass, I would like to thank you all again to be here in person or online, and also to thank everybody that worked very hard to make this happen. And special thanks for each one that make a presentation here, divide your work here with us. It was a great pleasure to know all your work, and it was a great contribution. I know that you spend a lot of time, dedicate time to be here and to share your work with us. It was very important, very productive. And I hope that next year we are going to have another event that we are going to see all the progress and the new ideas and new things that we are going to share here. Thank you very much, and I've finalized the event now.
Could I just ask all the Supergas Brass employees that are here, if they go in front of the checkerboard, or someone can ask the photographers to take a photograph of all the Supergas Brass employees. Thank you.